All right. Well, good morning, everyone, and welcome back to the 2022 Tropical Conservation Biology and Environmental Science Symposium. So this next session is going to focus on the amazing science being conducted by students and faculty right here at Hilo. Presentations will run until about 12:30 today, after which we're going to take a lunch break. So each presentation will be followed by a brief Q&A afterward. As a reminder to our presenters, without automatically advancing slides, either ask for next slide or give a nod so that the moderator knows to advance to the next slide. So starting us off today is Amberly Pigao with her presentation titled Optimizing Forest Restoration Technique to Increase Species Habitat and Mitigate Future Drought. Kanakaleonui Bird Corridor. Amberly, go ahead and take it away.
Mahalo, John. All right, aloha. My name is Amberly Pigao, and I'm from the island of Kauai. And I'm so happy to be here with you folks today and start off the 2022 TCBES Symposium. So I'll be sharing my thesis research on optimizing forest restoration techniques to increase endangered species habitat and mitigate future drought at Kanakaleonui Bird Corridor. Next slide, please. So my thesis is based on the east-facing slope of Mauna Kea, the highest mountain in Hawaii, and also a very sacred place to our people. Here sits the high elevation forest under a pu'u named Kanakaleonui. This place is very unique. It's a high elevation subalpine forest that has characteristics similar to a montane cloud forest. Kanakaleonui Bird Corridor, and I refer to it as KBC for the sake of time, is an enclosed fenced area of 514 acres. Here, cloud cover and fog presence are abundant daily. Unfortunately, this forest has been in a rapid decline due to the factors such as exotic species, both invasive grasses and ungulates, overgrazing, degraded soils, and harsh climate conditions. Added to these stressors are climate change, making it a threatened ecosystem of being permanently converted to degraded grasslands. KBC is very important due to its connectivity with the downslope Piha Forest Reserve, Hakalau Forest National Wildlife Refuge, and upslope Mauna Kea Forest Reserve. Establishing this corridor and connecting these forests will provide a path for native forest birds to move upward for critical survival and resistance to mosquitoes and the malarial parasite. Next slide, please. So the ability of KBC to capture fog has been degraded due to the loss of full-grown trees that would otherwise collect water on their leaves, subsequently dripping to the ground and percolating to the soil. Due to the land use history and current lack of water inputs, these areas have turned to resilient degraded grasslands. While seeds of dominant trees exist in the seed bank, these seeds are not germinating and are surviving due to degraded soils, which resulted from the lack of soil moisture, plus the lack of canopy cover. Next slide. So my research objectives are to capture moisture to water out planted seedlings. I have two separate experiments to see how we can most effectively facilitate survivalship. We are using 5 native species that once thrived in this dry forest, and they are Ave'aveo, Mamane, Na'e, Pavale, and A'ali'i. Next slide. For the two different experimental designs, native seedlings have been planted under nurse tree canopies and their drip line, and also next to shade cloths that are installed on fences. Both of these systems will capture moisture and protect seedlings from freezing temperatures. Soil probes that read moisture and temperature will take daily readings at each site, giving us information of abiotic variables. The more information we know about what's happening underground in this forest will help us better implement future planting strategies. Next slide, please. So for the nursery design, we selected fully matured nurse trees and planted 10 native seedlings, 2 of each species, under the drip line, and then 10 seedlings in each paired control plot. The nurse tree acts as an incubator to protect new seedlings by creating a wind and cold buffer, increasing shade, nutrients, and protection from herbivores. So the large circles represent the nurse tree canopy, the small colored circles represent the seedlings, and the rectangles represent the soil moisture probes that are buried in the ground to collect daily water content. The control plot is away from the nursery with a similar slope in an open area. Next slide. So the existing fence line is a perfect anchor to install the mesh cloth on because fog is lightweight and moves with the wind horizontally. The idea is that the mesh will capture the moisture when fog passes through the shade cloth. The moisture will then drip down to the ceilings that are planted alongside the mesh. The shade cloth design consists of 40% shade cloth, 63% shade cloth, and fence-only treatments. The small circles again represent the ceilings that are planted above and below the fence. Soil probes and temperature gauges will also be collecting daily levels. Next slide. Our experiment is located at 2 different elevational zones, approximately 7,000 feet and approximately 8,000 feet. The abiotic variables are different at both sites. We can see the amount of vegetation present. The upper site tends to be colder and drier and harder to grow plants, while the lower site has much more vegetation and moisture. Next slide. In 2021, we planted seedlings for the nurse plant design and the fence design. The upper site has 10 Mamane nurse trees with paired control plots, along with 10 3-part fence replicates. Again, those are 63%, 40%, and fence-only treatments as 1 replicate. The lower site has 10 Mamane nurse trees with 5 paired control plots and 10 Koa nurse trees with 5 paired control plots, and also has 6 3-part fence replicates. Next slide. Survival, growth, soil moisture, and temperature will be measured through a course of a year at nurse tree and fence replicate sites. Another set of data will be taken in the months of June and again in September of this year. We will be recording survivalship and of seedlings, looking for differences between the treatments of each experiment. We will see whether higher survivalship correlates with higher soil moisture and or air temperatures. Next slide. I am currently in data collection, and it's very interesting to see seedlings respond not only to the treatments, but to different areas of this forest. Here are a few photos of Availville, Naʻenaʻe, and Pavale adapting very well to this climate. Next slide. With this study, we will also learn characteristics of these native plants and how they are adapting to climate change and how they interact with the natural topography of this forest. Facilitating early stages of growth, we hope to outcompete the non-native grasses. Through this experiment, we can make assumptions as to which plants are best fit for Mauna Kea's conditions today in the face of climate change. Next slide. From our findings, we can observe and learn what the forest is telling us. Any information on this space and how these plant species interact with KBC's atmosphere will be useful and lead to greater survival of plants. The knowledge we gain from this 2-year study will benefit the land managers of Mauna Kea by adding information to their management strategies for native forest reestablishment. By restoring forests, we restore our resources and further perpetuate our Big Island communities. This study will also shed some light on KBC that is a very unique and important place to Hawaii. With these opportunities, to use nurse trees and shade cloth systems capturing fog water that could supplement the precious water resource necessary to reestablish trees, resulting in saving Mauna Kea from further decline. Pahai no ka'ua I ka'ulu la'au, which means the rain follows the forest. Final slide. So in closing, I would like to say a big mahalo to PICAST for this opportunity. the University of Hawaii at Hilo, my TCBES Ohana, my advisor, Jonathan Price, collaborators, Stephanie Yelenick and Corey Yanger with USGS, Cheyenne Perry with Mauna Kea Watershed Alliance, Kuali'i Kamara at Department of Fine Homelands, and also a big special mahalo to my friends who volunteered their time to plant hundreds of plants at KBC. I'm happy to take any questions. Mahalo for your time.
All right, guys, so we've got time for just a couple of questions here. You can either raise your hand if you want to ask directly, or you can throw it in the chat, and yeah, we'll ask them for you.
So we have a question in the chat. Do Kanakaleonui managers see this design as possible to replicate across the corridor at a large scale if results show promise? Yes, it's definitely would be a good way to facilitate growth at this forest. So yeah, it can definitely be implemented across the whole forest. Thank you, Scott.
Well, great question. Yeah, do we have any other questions that we can throw in? All right. We're going to give a big mahalo to Amberly here. We, I guess, need to keep moving. So again, thank you for the great presentation and for that awesome question. And we're going to pass it off to Noah Hunt here for his presentation, which is Nature's Playlist on Shuffle. Take it away, Noah.
Awesome, mahalo. Wherever you are, I'd like you to close your eyes for a few seconds and just listen. What do you hear? Can you imagine that at one point Hawaiian forests were filled with an almost deafening chorus of bird songs? Over 2/3 of Hawaii's endemic birds have disappeared since human arrival, the rate of extinction accelerating with Western colonization. By the early 20th century, Hawaiian residents noticed their forests had grown quieter. To replace their lost bird song, governments and private individuals released foreign bird species, many chosen specifically for their melodious songs. While most failed to establish, some succeeded to become the most abundant birds in Hawaiian lowland forests. Meanwhile, Hawaiian forest birds face threats from avian malaria, invasive predators, and habitat fragmentation and degradation. Yet so far, no one has studied how introduced species are influencing native bird communication. We know animals use signals to attract mates, defend territories, and warn others of danger. If introduced birds are competing with native birds for acoustic niches, they could be jamming their signals important to their survival and reproduction. What is an acoustic niche, you may ask? The acoustic niche hypothesis states that species have evolved in their vocalization to avoid interference from other species by adjusting the frequency and timing of their signals. In other words, if you and I were both birds, you would sing at a different frequency from me if we tended to sing at the same time. Or if we shared the same frequency, you would time your songs before, in between, or after my songs. We find examples in nature of colonizations generating acoustic competition between species. On Daphne Major of the Galapagos Islands, the medium groundfinch and cactus groundfinch overlap in range, but have distinct songs. In 1983, the large groundfinch, which sings within the same frequency range as the other two, colonized Daphne. Over 2 decades, the medium groundfinch and cactus groundfinches shifted the timing of their songs away from the new arrivals. These changes resulted from sons of both species singing faster than their fathers. Sons learned to respond to an exaggerated form of their own species' signal to avoid confusion from a closely related species. This demonstrates how behavioral traits can be passed down through generations in a similar way as genes. and maintain barriers between species to prevent hybridization. But what would happen if this barrier suddenly breaks down? On Kauai, 3 endemic honeycreepers have experienced decreased song diversity and complexity as a result of severe population declines. One species, the akekee, has also started to sound more similar to the other 2. This is probably because with fewer kumus to learn their songs from, young males incorporated songs from the other species into their repertoire. This brings up the question, could native birds also be incorporating elements from non-native bird songs? So far, there's no evidence of this. However, at least some songbirds can learn to adjust their timing to avoid interference from introduced species. For example, in Italy, blackcaps learned to sing in the intervals of songs of red-billed Leothrix, an introduced species. And while there are several studies of acoustic invasion from one species, there's been little investigation where sound communities have been colonized with many new vocalists. That's why Hawaii presents a unique opportunity to study this. Hakaua Forest National Wildlife Refuge is one of the most pristine montane rainforests in Hawaii with the highest endemic forest bird diversity. For my study, I'll conduct bird surveys in the refuge to determine relative abundance of native and introduced birds. I will then characterize the 3 avian community types, native dominant, mixed, and introduced dominant, and collect recordings from each with song meters. Then I'll select songs from all species in the recordings and measure their frequency, duration, and amplitude. Then I'll reshuffle the selected songs within each recording and measure their chance of overlap according to this randomization. I will then compare this with the actual observed song overlap, and there are 3 possible outcomes. If there's less overlap than random, introduced and native birds may be avoiding one another. If there's more overlap than random, they may be converging on sound space and competing. If overlap is equal to random, then interaction is unlikely. By playing nature's playlist on shuffle, I hope to determine whether Hawaiian birds have adapted quickly enough to find their own airtime, or if their broadcasts are being muffled by new songsters. What songs will we hear in our future forests? That's for us to decide. Mahalo for listening.
All right. Awesome. Yeah, that was so great. Thank you so much, Noah. So yeah, we have time for just a couple of questions here. Again, same thing. You can either throw them in the chat or you can raise your hand and we'll unmute you. All right, give it about just 30 more seconds. If we don't get any other questions, then we'll go ahead and move forward here. All right, so one just came up in the chat here. Really interesting investigation of soundscapes. Great job, Noah. Communicating effectively, such complex and exciting work. And yeah, I think that's all we're gonna have time for. We gotta keep moving here, got a pretty tight schedule. So
Might be off the hook. I think we've got one though. Does the bird song used for species classification, integration of other songs of native birds change their species classification? That's like a behavioral trait. It changes the culture of the animal, but it doesn't change their genes. So using a genetic definition of species, it wouldn't change their species. And I think I've got to get off the air. Thank you, guys.
All right, well, thanks so much, Noah, and thanks for that great question. Okay, so next up, we have Darrian Muraoka with understanding human-induced waste within stormwater runoff here in Hilo, Hawaii. Take it away, Darrian.
Mahalo, John. Aloha kakahiaka kako. My name is Darrian Muraoka. I am from Kekaha, Kauai, and today I will be presenting to you on understanding human-induced waste through stormwater runoff. Next slide, please. Many parts of this project that I will be presenting on are touched by various places within Hilo. Therefore, I would like to give my warmest aloha and mahalo to Hilo, the community, wahikupuna and nākua. Mahalo for letting me learn from you, about you, and continually allowing me to be a part of something bigger. Next slide. Before we start, I wanted to give an overview of what I will be talking about today. First, we'll be going over some background of my project, Waiʻola, the internship I'm a part of, my project, Kulana no I'i, and we will close. Next slide. Hilo evolved from a productive farming and fishing wahi into an industrial leader of sugar plantation within the 1800s. Downtown Hilo was built around Hilo Bay, and since then the county has managed the island primarily by dividing resources between the two most populated sites, Kona and Hilo. Next slide. Hilo is the largest city on Moku o Keawe with a population of approximately 45,000. However, since transitioning out of the plantation era, there are still relatively many agricultural plots and residences. In Hilo, we have our rain, Kanilehua. As it rains harder, a lot of pollution runs from the town and into our river sources, which ultimately ends in Hilo Bay. Because of the infrastructure of the breakwall, a lot of human-induced pollution has collected and settled into the sediment and altered the ecosystem around it. Next slide. So what is impacting the water quality within Hilo Bay? We have stormwater runoff. As a result of young geology, our city experiences intense runoff, which flows down into Hilo Bay and becomes trapped by the breakwall. We have discharge from municipal facilities. The water quality in this area is especially compromised by a variety of municipal pollution sources, which have all been seen by our little small town. The related pollution especially affects many people in the area who continue to rely on subsistence fishing traditions to feed and support their families. Water infrastructure, there is only one municipal sewage treatment facility in the Hilo district, and it is located on the Hilo Bay in Keaukaha. Many swimmable and recreatable beaches are within the same community, and these beaches have been closed multiple times in recent years because of high levels of bacteria. We also have public awareness and education. People recreate, fish, and gather at these coastlines, and many residences are familiar with legacy pollution and know how much the rain can change the area. We need to make sure that everyone knows about the long-term health effects to Kanaka and Vai and all these places we want to enjoy. Next slide. And so with this in mind, Waiʻola Ohana was born, the nonprofit I'm interning under. We started as a chapter in the Waterkeeper Alliance, a global movement to protect water resources, and we have recently rebranded our now called Waiʻola Ohana and taken these sub-chapters and created them into one big ohana. We are still working under the goal of swimmable, fishable, drinkable waters. Through our mission, we are continuing efforts to make sure our waters will not be impaired for future generations with the County of Hawaii and the EPA. Next slide, please. With this internship, the principles of Kulana no I'i and its award helped me fund this project and gave me the opportunity to reevaluate the research I am doing from a different lens and to see how our resource stewards, community members, and I could connect in significant ways, which closes the gap of working with the community and helped me build Kulina, to commit to it and aloha aku, aloha mai. Love outward, love inward, love everyone and everything. Next slide. And through this award and collaborations with the 2 years I have, this really got me excited, and I wanted to look at human-induced pollution through many ways by drone footage, water quality monitoring, and outreach and education. Doing this was sealing that we will be dealing with the 4 main impacts of the Hilo watershed. Next slide. We got into contact with Hilo's own Andrew Hara, a National Geographic visual media specialist, and we told him our vision of wanting to capture our 3 main rivers. We wanted him to go out into the field after big rain events to capture the power of Vai and where that Vai flowed within the watershed. So he captured Wailoa, Wailuku, and Honolii. Next slide, please. And please play the video. This is a clip of the mouth of Honolii, a very amazing place to hit in Nalu. After a big rainstorm, we don't see much of the bay or the river mouth that holds the river in and creates a beach, we see a change in water color extended into the ocean, an open river mouth, and a pretty small lineup. Next slide, please. The next slide, we see Wailuku. Here we have Wailuku looking over Waianuenue Falls during the big rain event. The power of Wai is ever so present here, bringing down trees that floated in a pocket at the base of the waterfall. We also see some residences, businesses, and farms that could be leaching non-point source pollution in Wailuku, which will ultimately lead into Hilo Bay. Next slide, please. And please play the video. Last, we see a part of Wailoa River before it empties into the bay. On what looks like a clear day, we see a lot of residential areas and businesses still sitting on the banks and around Wailoa River, which can tell us where non-point source pollution is coming from. The next step in this part of the project is to take the full video and find on our County of Hawaii storm drain and sewer line map where cesspool leaching and storm drains are emptying out in order to update the county on what our impacts are within these rivers and what we can do to filter toxins before they reach the bay or ocean ecosystems. Next slide. The other side of the project is water quality testing. 2 days after a big rain event, we tested water from our sites at the headwaters of our rivers, a randomized point in Hilo Bay, and the shore near the outflow pipe in the beginning of Keaukaha. Next slide. So what are we testing? I am actively testing for 3 big things: turbidity, to see how suspended the particles of sediment were in the water; optical brighteners, a brightening chemical compound found in detergents; and metals such as arsenic, lead, and mercury. We are actively testing for these agents, so I do not have conclusive results yet. However, check back in the fall for the full report. Next slide. The last part of this project was outreach and education. This is my favorite, as I got to talk story with community members, volunteer at places I've never been to, and create those connections and values that Kulana no I'i has helped to instill. With this part of the project, I initiated a storm drain stenciling campaign, helped in collaborative efforts with nonprofit organizations, and work with amazing students. Next slide. For a public awareness campaign, I initiated storm drain stenciling with the Department of Public Works. We confirmed spaces with the county for circled here as accurate places within Hilo, where the storm drains do lead to nearby rivers or out into Hilo Bay. I took Waiakea High School students to downtown Hilo to take pictures of the storm drains before stenciling and to take data on it, where it was, if it had recently collected any pollution. And we will be starting storm drain stenciling in the weeks to come and also this summer. Next slide. For Earth Month, we created a podcast with Maestro's Vibe, a Department of Education based in Konowena podcast that talks about things going on within our community, Moku o Keawe. I was on the first episode and talked about stormwater runoff and awareness campaigns. Next slide. Last week, we collaborated with Kanu Hawaii, a nonprofit organization, to create volunteer opportunities during the week of Earth Day. One of the sponsors was Ace Hardware, and we got the opportunity to create cleanup kits and distribute it out to all the Ace Hardwares on Moku o Keawe for people to start their own cleanup groups and to celebrate their spaces. On Earth Day, we gathered with the mayor at the bandstand in Hilo and the county council member for District 4, Ashley Kirkwitz. We chanted E Ala E, we brought up the sun, and had the mayor proclaim Earth Day. We then ended the morning with a coastal cleanup in downtown Hilo with many volunteers from the community of Hilo. Last slide, please. Next slide. Lastly, I collaborated with Hui Ho'ole Mauluo and Lanakila Learning Center to teach students about water quality within the fish pond and how to create opportunities for pono science. So within Kulana no I'i, we built Kalina by reaching out to our Hui Ho'ole Mauluo. We communicated, talked stories, learned about Kaumaui and about Keaukaha. And then we nurtured our Kalina by coming back. And that's the biggest thing, making new connections and committing to those connections. And lastly, we engaged to set dates for sampling, presenting to Lanakila Learning Center, and also to have the students come out to the fish pond. Next slide. And so our day at Kaumaui, we did protocol. So we did our chant, paid respects, and opened the day. We next did Hilo. So we connected with the place, interactions with the environment, and we dove into the wonder that was our natural and physical world and made sure we were ready for the day. Next, we did pH and turbidity to teach students how to take water samples. And last, we holo, so we caught some tilapia and male. Next slide, please. This is just examples of how we did Hilo for that day. So we did an observation log to collect qualitative data. And so we got to know what the moon phase was. How are we today? Our winds, our clouds, and also if it was raining and if it was high or low tide. We also did observational logs of what is in our marine environment and also our terrestrial environment. So we had all of this going on while we kiloed in the morning of that day. Next. Next, we took turbidity of the makaha. So the turbidity at the time of sampling was low, which means little to no particles were suspended in the sample of water we took from the makaha. Next slide. We also took the pH of the ponds. I then averaged them to see if there were any drastic changes within the ponds themselves, and I found they were all neutral, which is a good sign. Next slide, please. Last, we holo and caught different types of fish found in the pond. We caught male, o'opu, and tilapia. With the male and invasive tilapia, we opened up the stomach content to see what the fish were eating, and then we took the tissue samples to analyze in the lab to look for any traces of metals. We gave the fish to the students and their family to eat for that night. And this, what we will find will add to the baseline data for Kaumaui to see if these ponds are able to raise fish stocks and give to the community or sell in the local markets. Next slide. Overall, this project has progressed immensely and it wouldn't have been made possible without the help, hands, ike of many. I want to thank you for your time and take any questions you have. Mahalo for listening and mahalo again to Hilo and its people. Mahalo, Pika.
Okay, mahalo, Darrian. So thank you so much for that excellent presentation. For the sake of time, we're running just a little behind. So if you can go ahead and put your questions in the chat, we'll let Darrian answer those on her own independently, and we're going to keep on moving. So next up, we have Rachel Willard with shell utilization patterns in Hawaiian hermit crabs, choice or availability. So Rachel, go ahead and take it away.
Thanks, John. All right, aloha, everyone. I am Rachel Willard, and my senior thesis here at the University of Hawaii at Hilo has been dedicated to shell utilization patterns in Hawaiian hermit crabs. Is it choice or availability? Next slide. So hermit crabs are arthropods under order Decapoda, superfamily Paguroidea. They are not considered true crabs and are characterized by their soft abdomens and tails, resulting in their reliance on empty gastropod shells or other hollow objects as a means of protection. There are over 800 species of hermit crabs worldwide and approximately 23 species around the island of Hawaii. Hermit crabs here are abundant in tide pools and on shallow reefs. Hermit crabs do play an important role in the benthic community. They are omnivorous scavengers, recycling energy back into their ecosystems. The unique lifestyle of a hermit crab is particularly successful due to the mobility and protection that a shell offers. Utilizing a shell offers them protection from predators and other environmental stressors and the ability to roam freely. Different parameters of a shell's fit determine a hermit crab's growth, survival, and fecundity rates. Hermit crabs occupy their shell until they grow larger and will then need to seek a more accommodating home. In some habitats like Sanibel Island off Florida, for example, there is a vast abundance of gastropod shells, and hermit crabs there have the ability to be opportunistic with their shell selection. Here in Hawaii, shells are a limited resource and hermit crabs will occupy whatever shell is available to them. Hermit crabs will find empty shells or participate in shell exchanges. Sometimes only poorly fitting or damaged shells are available, but for a hermit crab, even poor protection is better than none. Next slide. As hermit crabs around Hawaii tend to be shell limited, competition increases and shell fit decreases. The intention of my research was to determine which species of gastropods and hermit crabs are common around the island of Hawaii and what species of shells local hermit crabs occupy. It was hypothesized that one, gastropod shells occupied by hermit crabs reflect the abundance of gastropods in the habitat. Two, similar sized hermit crabs utilize the same species of gastropod shells. And three, same species of hermit crabs utilize the same species of gastropod shells. Next slide. To determine which shells are available for hermit crabs around the island of Hawaii to utilize, all gastropod shells were collected in 5 by 5 meter quadrats in 0 to 10 feet of water for snorkeling and 10 to 20 feet for scuba. Quadrats were sampled at different beaches around the island beginning in October last year through March this year. All full or occupiable gastropod shells were collected, regardless of current occupancy being a snail, hermit crab, or empty. All collected gastropod shells were brought back to the beach and placed in buckets of salt water to be identified and measured. Gastropod shells were identified to species when possible and measured with the ruler in centimeters. Current occupancy of the shell was noted. Hermit crabs were also identified to species level and placed in a size category. All shells and hermit crabs were then released back near where they'd been found. Next slide. Quadrats were sampled at 4 different locations around Big Island, including Richardson's and Onekahakaha, both in Hilo, and at Manini Beach and Two-Step in Captain Cook. Next slide. In total, 628 shells were collected. This included 17 gastropod families and 9 hermit crab species. 46% of the shells were found occupied by hermit crabs, 37% by gastropods, and 17% were found empty. Families Cerithiidae, Conidae, and Neritidae were the most notable families occupied by hermit crabs. Next slide. In the results of my analysis, I did find that hermit crabs around Hawaii Island do occupy shells that reflect the abundance in their environment with a p-value of 0.001. Overall, similar sized and same species hermit crabs utilizing the same species of shells did not show to be true, although a preference was found to be significant, specifically for hermit crab species Calcinus latens, Ciliopagarus strigatus, and hermit crabs categorized as size large. The other species and sizes with an insignificant p-value are selecting their shells based on availability and not preference, concluding that Hawaii is shell limited for hermit crabs. Next slide. I would like to thank Carla and Marta for advising me, Edit Karth for coordinating equipment, and all the other students involved at the university for helping in my project. Next slide. And if there's time, thank you all, and I can take any questions.
Yeah, thank you for that, Rachel. Yeah, so we have time for maybe 1 or 2 questions here. Again, go ahead and throw them in the chat or raise your hand and we'll go ahead and call on you. Give it about 30 more seconds, and then if we don't have any other questions, we'll go ahead and move on.
Okay, I used a chi-square test. Give a p-value. Thank you.
Awesome. All right, well, thank you so much, Rachel. We're gonna go ahead and keep moving here. So next up is Ihilani Kamau, and with his project titled Effects of Stocking Density and Nutrient Treatments on Growth in the Native Hawaiian Red Macroalgae Lepe-o-hina in Culture. All right. Ihilani, go ahead and take it away.
Aloha mai kākou. My name is Ihilani, and today I will be talking about the effects of stocking density and nutrient applications on the growth of the native Hawaiian red macroalga Lepe-o-hina, or Halymenia hawaiiana in culture. Next slide. Yeah. So Halymenia hawaiiana is a Hawaiian Limu species. It comes from the red algal genus Halymenia. They can have a variety of colors ranging from red to yellow. And something interesting about this species is that when it was first described, it was actually called Halymenia formosa. So if you look back into previous studies, a lot of the times it would have the Hawaiian name Lepe-o-hina, but the scientific name would be Halymenia formosa. So after being reassessed and reanalyzed based on a DNA analysis in 2012 by Hernandez Cantun, they found that this species of Limu was actually a new species, and then that's where they called it Halymenia hawaiiana. Next slide. So here is an image of Halymenia hawaiiana, and it can be described as having large branch thallus, flat blades, irregular margins, and soft spinose proliferations or ruffles. So its Hawaiian name, as I said earlier, is Lepe-o-hina, and that is like the shawl of Hina. So something interesting about the species is when it's in the water, it kind of moves, like a lot of people reference it as like a pahu skirt when you're dancing. So it flows really, it's really flowy like a pahu skirt, and the ruffles really bring that out when it's in the water. Next slide. So why did we choose Halymenia hawaiiana? So it is a native species in Hawaii, to Hawaiian waters, and as a Hawaiian, it's important to understand the limu that's growing around you for its cultural importances, nutritional benefits, as well as the roles that it can play in your ecosystem. It is also an edible species. In a study done by McDermott and Stork in 2003, they found that Halymenia hawaiiana has a high protein content. The main reason why we chose this species for our experiment is that there's not really much known about Halymenia hawaiiana or about its growth. So we're hoping that by doing this experiment, more people will know about this species and how it grows, especially in culture. Next slide. So the two research questions that I focused on in this experiment is, what is the optimal stocking density or the optimal initial biomass for the growth of Halymenia hawaiiana species in culture? And the second one is, what is the optimal nutrient application for the growth of the Halymenia hawaiiana species in culture? So the two main things I was focusing on was finding the optimal stocking density as well as the optimal nutrient application. Next slide. So for my experiment, I did it at the Pacific Aquaculture and Coastal Resource Center in Keaukaha. So on the top left, you can see like an aerial view of the site, and I put a little star of the exact location where I did this experiment. And on the right, you can see the tent, and that is basically where my experiments took place and where all of my buckets were held throughout the duration of this experiment. Next slide. Next slide. Oh yeah, okay. So for the stocking density setup, I had 12 19-liter buckets. So I was able to have 3 replicates. All of the buckets were supplied with a fresh flow of seawater, also aeration through those little clear tubes. And all of the buckets were covered with 2 layers of 30% shade cloth, just to ensure that they weren't getting too much sunlight. And since this is a stocking density experiment, the densities that I focused on was low, medium, high, and high. So for low, I had 2 grams per liter, medium, I had 4 grams per liter, high, I had 8 grams per liter, and high, I had 18 grams per liter. So after all these buckets were set up, I basically weighed out all these different densities and distributed them randomly throughout the buckets and just labeled the buckets based on their densities. Next slide. So for this experiment, the most important data that we collected was the weight, because we're looking at the growth in this experiment. So we weighed our samples once a week, and it was a really simple method by just taking the limu out of their buckets placing them into a salad spinner and spinning them for about 30 seconds. And then after that, to just dry off the excess water, we used a towel and placed it into a container that was on a scale that was already tared out, and just documented, I wrote down what the weights were. Next slide. Some other measurements that we took was we used a YSI to check the water quality of the buckets. We collected that once a week. We also used a light meter to measure the amount of sunlight that the Limu were getting, and we collected that twice a week. We also looked at the nutrient levels, so we looked at ammonia, phosphate, and nitrate levels, and that was also measured once a week. Next slide. So here's a figure showing all the data for the stocking density experiment. So on the y-axis, you can see the specific growth rate, and on the x-axis, that's the different weeks. And on the top right, there's an equation for specific growth rate, and that's a well-known equation used for to calculate the specific growth rate. So just looking at this figure, that you can see that in week 2, 3, and 4, the low density actually had the high specific growth rate overall. However, in week 1, the medium density had the highest specific growth rate. But it was interesting to see that the low density and medium density had the highest, while the high density and the high density had the lowest. So when looking at this figure, you can say that the low density had the high specific growth rate overall, and the high density, which was the buckets with the most biomass, you can see it consistently had the lowest specific growth rate. Next slide. So for the nutrient experiment, the setup was really similar to the density experiment, but instead of 12 buckets, we had 9 19-liter buckets. So we were still able to have 3 replicates. All of the buckets were supplied with a flow of seawater and also aeration. And instead of 2 layers of shade cloth, we had 1 layer 70% shade cloth. And in our previous experiment, we found that the stocking density was, the optimal stocking density was at low density. So carrying on to the nutrient experiment, we used the low density throughout all of the buckets. So all the buckets were filled with the 2 grams per liter of the Halymenia hawaiiana, and the only difference was the application of nutrients. So we focused on 10 milliliters of the fertilizer once a week, 10 milliliters of the fertilizer twice a week, and 20 milliliters of the fertilizer twice a week as well. And we just labeled that throughout the buckets. Next slide. So for this figure, it is the data from the nutrients experiment. So the with the axis having specific growth rate and the x-axis having the different weeks. And we also use the same equation for the specific growth rate seen at the top right. So as you can see in week 1 and week 3, the 10.2 or the buckets that are receiving 10 milliliters of the fertilizer twice a week seem to have the highest specific growth rate overall, while in week 2, the 10 milliliters once a week had the highest. But overall, the 10 milliliters twice a week had the highest specific growth rate. And just looking at this figure, you can see that there's like no real significant difference, but we found that the 10 milliliters twice a week had the best specific growth rate overall. Next slide. So in conclusion, for the stocking density experiment, we found that the specific growth rate of Halymenia hawaiiana was the highest at the low density of 2 grams per liter. And in the nutrients experiment, we found that the specific growth rate of the Halymenia hawaiiana was the highest with the 10 milliliters twice a week. But overall, there was no significant difference among the treatments. So you could say that the optimal culture conditions for the Halymenia hawaiiana appear to be at a low density and with nutrients being provided at a lower concentration post twice a week. Next slide, please. I would like to thank Rose Christian, Dr. Carla McDermott, and the Pacific Aquaculture and Coastal Research Center. Mahalo nui loa.
All right, mahalo Ihilani, that was awesome. It's so great to see some work that's done on species that we just don't know that much about here in Hawaii. So we do have time for a few questions. Also, for the sake of keeping things moving, if you guys want to start putting questions in the chat as people are presenting, I think that's a great way to keep things moving as well. So yeah, let's go ahead and see what we get here. All right, let's take just about 1 or 2 more minutes. Okay, guys, so let's go ahead and keep moving. Ihilani, again, thank you so much for your awesome presentation. And we're going to pass it off to Lily Gavigan with her presentation on investigations of unknown Limu, Annelida Polychaeta through microscopic morphology and untargeted metabolics via LCMS. All right, take it away, Lily.
All right, aloha kakayaka kako, everybody. My name is Lily Gavigan, and thank you so much, John, for the wonderful introduction. My name is Lily Gavigan, and I'm so honored to be here with you folks today to present a little bit about my thesis project investigating polychaetas using scanning electron microscopy and metabolomics. Next slide, please. So to give you guys a little bit of a background, Dr. McDermott and I found a large amount of Caulopa lentifera from the Mokupapapa Discovery Center. And within this algae, there is a lot of these little critters running around inside, as you can see on the bottom right-hand corner. And next slide, please. And so when we got a closer look at these worms, we saw that they were round and segmented. So we knew that they were from the phylum Annelida. And then we looked at the sides of them, and we saw that there was bristles coming out of them. And so we knew that they were bristle worms, which is also known as Polychaeta or ahahuluhulu in Hawaiian. And we knew from their bright color features, as well as unfortunately getting our hands on them and stinging a little bit, we could kind of conclude that they're from the family Amphinomidae, which are fireworms. And in Hawaii, we have 3 different genera of them, which is Eurythura species, Sphaerocardia species, and Hermodice species. Next slide, please. And so in this project in particular, we came up with a list of questions that we wanted to figure out, because we never really saw these worms before. So we wanted to figure out what was the genus of these worms based off of their morphological traits, as well as genetic sequencing. What is their growth rate? What do they eat? And if there's a significant difference in chemical compounds found between the worms, the limu, the seaweed, and the seaweed exudate, which is basically secretions on the surface of the limu itself. Next slide, please. So to do this, we initially wanted to run a morphological analysis on them. So we gathered some worms from the tank and we preserved them in a formaldehyde solution. And then we also ran them through a general microscope, dissecting microscope, to see what they really were. And this is where we found out that these were definitely polychaetas and bristle worms. And so after this general identification, we fixed them in a sodium chalcodilate solution to dehydrate them. Next slide, please. And then we further dehydrated them by running them through a critical point drying unit and then sputter coating them with gold. And then we were able to upload them to the scanning electron microscope and this is found on the Hilo campus in the Marine Science Building. Next slide, please. And so these were some of the images that we got, and this is what we also used to measure them in the initial phase of this project. So as you can see, we had a hard time getting them to lay flat. So it's a little difficult to get measurements on them. Next slide, please. Nevertheless, we were able to get length measurements, and we were able to calculate the specific growth rate from samples taken between the months of September and November. And from this calculation, we concluded that there was a 4.6% growth increase per day. Next slide, please. So since they're growing, we know that they're probably eating something in the tank. So we wanted to know if it was either the limu itself or if there was an exudate on the limu itself that they were feasting on. And so this is where we turn to metabolomics, the study of metabolites and what is in the organisms itself. So we collected samples of the worms, the limu and the limu exudate, we freeze-dried them, and then we grinded them and extracted them via methanol extraction. Next slide, please. And then we dried and further reduced the volume so that we can reconstitute it later. And in the future, we are planning to upload them to the liquid chromatography mass spectroscopy unit that you see on the right here. That's located in Puna'ewa at the College of Pharmacy. And so this is where we analyze the different chemical compounds of the worms, the limu, and the limu exudate. Next slide, please. So overall, with this project, we wanted to figure out if these worms that we see in this tank are cryptic species, meaning if these species are a different species that we're seeing compared to the species that we see in the wild. And we're doing this through genetic analysis in the future by genetically sequencing them. And we're partnering up with people at the University of Alabama to get a go ahead and figuring out what species these are. And we also hope to key them down to species by also trying morphological variations, looking at their morphology. And by doing metabolomics, we're also trying to figure out their overall feeding mode and thus their overall ecological niche in this ecosystem. Next slide, please. And I'd like to extend a huge mahalo to everybody on the screen who helped me get this far. And if we have time for questions, I'm happy to answer any of them. If we don't, I also located my email here on the bottom right-hand corner. And mahalo ya oko no ko'olohe ana mai. Thank you for listening.
All right. Well, big mahalo to Lily. That was awesome. Yeah, we do have a minute or two for a couple questions here. So again, go ahead and throw it in the chat or raise your hand and we'll call on you. And yeah.
Oh, thank you.
All right, we'll give it about 30 more seconds, see if we get any questions or just people that are generally pumped on your research. That's great too.
Oh, thank you, guys.
I mean, no questions so far, but a lot of good vibes. So that's positive. Okay, all right. Well, I think we're gonna go ahead and move on here. Again, if you guys do have questions as we go, just put them in, type them in the chat, and we can answer them as we go as well. So again, thank you, Lily, for that. Next up, we have Nikola Rodriguez with how will mass bleaching events affect the prevalence of ciguatera on Hawaiian reefs. So Nikola, go ahead and take it away.
Thank you so much for the great introduction. So I'll just go ahead and start on the next slide and jump right into it. So ciguatera fish poisoning is considered one of the most common marine toxin related illnesses in the tropics and now throughout the subtropics due to climate change. Accounts of illnesses consistent with ciguatera fish poisoning have been reported as far back as Alexander the Great in the early 300s BC. During his conquest, his soldiers were prohibited from eating fish to avoid getting sick. While rarely fatal, ciguatera fish poisoning is characterized by gastrointestinal, cardiovascular, and neurological symptoms, ranging from mild and short-term to debilitating and long-term, lasting months to potentially years. Some common symptoms are the reversal of hot and cold sensation, diarrhea, vomiting, hypertension, and sensations of pins and needles. Next. The effects of ciguatera fish poisoning are multifaceted and have extensive economic and ecological impacts. Hawaii had a ciguatera scare in the 1980s involving a lua, which led to a 90% decline in the commercial landings of this fish. This fishery has never recovered. Here is an article published in 2018 talking about an ulua that was caught by fishermen off the Kohala Coast, which was the cause of multiple ciguatera fish poisoning cases. This illustrates that still today, ciguatera fish poisoning is a concern in our local fishing communities. Furthermore, the fear of contracting ciguatera is the main factor which has led to the prevention, or which has prevented the development of the peacock grouper or roy fishery here in Hawaii. Next. Ciguatera fish poisoning is caused by the consumption of reef fishes containing toxins produced by dinoflagellates in the genus Gambierdiscus. Some of the reef fishes that are known to cause ciguatera fish poisoning in humans are jacks, barracudas, groupers, snappers, and sturgeons. These fishes have bioaccumulated naturally occurring ciguatoxins through the food chain. Next. On coral reefs with low herbivore abundance, Gambierdiscus densities have been observed to increase over 300% from their initial densities, inferring that the aerial density of Gambierdiscus may be affected by changes in herbivore fish's feeding habits, selectivity, and grazing rates. The increased availability of macroalgae due to the decreased rate of grazing provides a greater availability of suitable substrate for Gambierdiscus to colonize on and grow. Next slide. This infogram illustrates how ciguatera is thought to get into the food chain. The chain begins with herbivorous fishes incidentally consuming dinoflagellates and their toxins by eating the limu or algae. The toxins then accumulate in their flesh and they're later consumed by larger fishes, where their toxin is then bioaccumulated and biomagnified in the fish's flesh. Then in turn, those larger fishes are consumed by humans. Next. Climate change has been the cause of mass bleaching events worldwide, which provides fresh habitat for algae to colonize, in turn, providing new habitat to be colonized by dinoflagellates. Furthermore, at a more local level, climate change has the potential to alter Gambierdiscus prevalence and the risk of ciguatera fish poisoning, but this has not been thoroughly investigated. Next. Hawaiian reefs produce around 7 million meals per year. This makes ciguatera fish poisoning an economic, food security, and public health issue. By understanding how the prevalence of fishes with ciguatoxins is impacted by changing reefscapes associated with anthropogenic climate change is critical for the development of management strategies, such as the Sustainable Hawaii Initiative, also known as the Marine 30 by 30. For example, areas with high prevalence of ciguatera could potentially be new locations for marine protected areas. This information could allow state agencies and fishing communities to better warn the public about where the risk of ciguatera fish poisoning might be elevated. Next. So this brings me to my project objective. My first objective is to assess the prevalence of ciguatoxins in reef fishes along a disturbance gradient along the west coast of Hawaii Island. These gradients are caused by coral bleaching and herbivore abundance. My second objective is to assess the perceived risk of ciguatera fish poisoning in local fishing communities compared to the actual risk. Next slide, please. So my study focuses on two species, the peacock grouper and the golden-eye bristle-toothed tang, locally known as the roy and kole. Roy was selected because it is an introduced high trophic level predator that is thought to be high in ciguatoxins, so it is not taken for sustenance fishing. Kole is a small-bodied herbivore that is commonly targeted by commercial, subsistence, and recreational fishers. Each fish will be tested for ciguatera using test kits provided by CTOT. I will analyze the prevalence of ciguatoxins and related to coral cover and herbivore biomass. To date, 136 fishes have been collected, 58 roy and 78 kole. Each fish has been geotagged and otoliths have been removed for aging. I'll be mapping the location of each of the collected samples to collect habitat data for the fishes. And I'm also speaking with local fishers to assess their perceived risk of ciguatera fish poisoning in our local fishing communities. Next. My study takes place along the west side of Hawaii Island. My collection sites were chosen based on percent coral cover and adult herbivore biomass. This is to see if the differences in herbivore biomass and coral cover affect the prevalence of ciguatoxins in these collection areas. For my results today, I'm going to just give a brief summary of the fish collected and focus more on the perceived risk of ciguatera on our fishing communities. This map, at the very top, we have Pu'oko, and at the very bottom, we have Ka. So the percent coral cover at the top is about 4% at Pu'oko, and then the largest coral cover we have is towards the south in Ka. And then Anaeho'omalu Bay in the middle and Halipa'o, also known as Papavai, they both have around 14% coral cover. And then I have my adult herbivore biomass, which ranges between 4,900 in the south to about 12,000 grams per 100 meters square in the center of the areas where I'm collecting. So go ahead, next slide. Okay, so for all my collection sites, the mean fish sample sizes are similar for both Roy and Coley, which is a good thing. Here we're looking at the Roy fish. From Pu'oko to Ka, the mean length of the fishes collected range between 27.9 centimeters in the south at Ka to 34.5 centimeters in the north in Pu'oko. Next slide. Again, for Coley, there is an average length is similar as well for each location, which again is good. The Pu'oko in the north, the mean length is about 15 centimeters, 15.1 centimeters, and all the way in the south is 15.2 centimeters. Next slide. So now I'm going to get into the perceived risk of ciguatera fish poisoning in our local fishing communities. I spoke with about 27 fishers and divers and asked them about, how they've learned about ciguatera fish poisoning, what factors do they think increase their risk of getting ciguatera, what do they do to prevent themselves from getting sick. When I talked to them about where they learned about ciguatera, 25% of their answers were ohana, their family members. 17% of them were either divers or fishers, and another 17% said the intranet. I was surprised to hear only about 8% said scientists or scientific journals and TV. So hopefully we can get those numbers up a little bit. Next slide. Then I asked what factors they feel increase their risk of contracting ciguatera fish poisoning. I got a wide range of answers, but location was the main factor that people thought increased their risk. 23% people said location by resorts, golf courses, high population areas. 19% said the fish species was a factor. They wouldn't eat roy, they didn't fish for ulua, more the larger predators they kind of stayed away from. 3 other factors that stood out were the amount of fish, so consumption, and what part of the fish they ate, like the head and the belly. They said to stay away from those. And then of course, the size of the fish too, was those 3 were kind of all in par at about 12%. And then the remaining factors across the board were about 4% each. Next slide. When I talked to fishers about what they actually do to prevent themselves from getting ciguatera fish poisoning, I was really surprised by their answers. 25% of the fishers I talked to said they just chance them. They believe that the only way of not getting ciguatera fish poisoning is by not eating reef fishes at all. So they just chance it. 19% say they only eat smaller predators. 13% said they only eat fish from specific locations. 13% said they eat specific fish in moderation if it's thought to be high risk. Some of the other ways that weren't so popular were they feed it to the cat first, see what the cat, if it eats it or if it doesn't survive after eating it. One is they only eat manpati or veo, and the other one is don't eat reef fishes at all, or they just don't eat the head and body. Next slide. So in conclusion, for my first objective, the sample collections across the sites, which are similar across the site, which is good because my results should be telling about ciguatera prevalence for each location. For my second objective, with the preliminary data that I've collected, I find that fishers and divers are going to eat fish no matter what. The ocean is their refrigerator. Their perceived risks and thoughts on how to mitigate catching or contracting ciguatera fish poisoning is in line with my project objectives. I believe that my results will be able to help them know where the risk of ciguatera is greater with changing reefscapes due to climate change. Next slide. So my next steps for my thesis are going to get all the tissue samples I've collected screened for ciguatoxins, age the fishes, map the collection areas and analyze that habitat data, and then continue to assess the perceived risk of ciguatera fish poisoning in our fishing community. Next slide. I would like to say a big mahalo to the Pacific Island Climate Adaptation Science Center and to the U.S. Fish and Wildlife Center. My project is jointly funded by PICAS through UH Hilo and the U.S. Fish and Wildlife Services. Without them, my project would not be possible. And a big mahalo to my committee and to DAR for helping me facilitate my project. And with that, I'll take any questions.
Awesome, that was great, Nikola. So we already have one question that's a super interesting one here. It says, I actually caught and ate some roy a few years ago when I didn't know about ciguatera. Is it possible I have ciguatera dormant in my system?
Well, ciguatera is known to be metabolized. However, there's not a whole lot that people know about it. So it's thought that the more fish you consume, whether it be reef fish or whichever, then the higher your natural ciguatera levels, I guess not natural, but your accumulated ciguatera levels would be. But I don't know how long it takes for the body to actually metabolize those out. So how long they would stay in your body for.
Yeah, super intriguing. So I wanted to make sure I connected some dots here too. I'm also just going to start jumping into questions while people are putting them in as well. So I wanted to make sure I got this right. So the idea is that as coral bleaching increases, that would increase the amount of algae. And because algae often is the source of ciguatera initially, that would lead to maybe higher levels of ciguatoxin in fish generally, or just in a handful of these species. Did I get that right?
So the algae doesn't actually cause the ciguatoxins. It's the dinoflagellate that lives on the algae. So it really just depends on the gamba discus, like if it's present or not. And there have been certain studies that, you know, look at wave action and whether wave action influences the prevalence of the gamba discus in the algae, and whether location is a difference. But with climate change and bleaching, if there are more bleaching events, there is more available substrate for the algae to grow, which then again creates more habitat for the gamba discus.
Cool. All right, good to know. And yeah, we've got a few other questions here. So it says, the community engagement and community perception elements within this work are super exciting. Were your connections to community fishers already existing prior to starting this project? And if so, have you grown new community connections through your exciting interviews or exiting interviews, whichever one?
Yes, I do have quite a few friends that are Spiros and divers, and I'm a diver myself, and my partner is a fisher. And so I did have a little bit of a connection to the community, but just with, you know, getting onto these different, I guess, social media sites and inquiring and getting people to actually help out with my project, because 99% of my fish have been collected by other people, not by myself, which is, I'm very thankful for everyone who has helped me collect fishes, because 136 fish with 58 roys is definitely a feat in itself. But yeah, I've definitely grown my spearfishing community, and as well as people who are kind of really interested in this project and to see what the results are. And, you know, because like I said, it's their refrigerator. So they're kind of interested to see if there's all these bleaching events happening, if, you know, they might be more of a risk of the ciguatoxins in their fishes.
Awesome. Yeah, we have one more question and then we'll go ahead and move on here. So Pat Hart asked, said, neat study, and is there any easy way for fishers to test their fish for ciguatoxin?
Not currently that I'm aware of. I know there used to be a kind of at-home test kit that was sold at some of the fishing shops, but I think it had too many false positives or false negatives that they removed it eventually. So I'm actually using a commercially available one but that I actually have to use at the College of Pharmacy's lab to run it all. So.
Oh, wow. So you have to actually use lab equipment to test your ciguatoxin right now.
Yeah.
You know, awesome. Okay, well, great. Thanks for all those awesome questions and great job, Nikola. So we're going to go ahead and move on to our next presenter here.
Thank you.
So let me introduce Nathaniel Greenwald and his presentation titled "How Many Ama Are in Koloko?" All right.
Awesome. Thank you. Should I share screen with it? Oh, there we go. Awesome. Thank you. Next slide, please. So today I'll be talking about how many ama there are. And so in Koloko, and Koloko is a 4.5 acre fish pond currently managed by the Park Services. And efforts to restore the fish pond and native vegetation have been undergoing. But through the community, there have been strong desires to restore the fish production for harvest and or provide nursery habitat for the surrounding fish populations. Next slide, please. So the needs and realities. So the national parks really need something repeatable and scientific-based monitoring capable of generating population estimates in high confidence intervals. And they also want to protect endangered species and of bird species and leave undisturbed, since this is a native nesting ground for them. They also want the silt of the bottom of the pond to be left undisturbed, so if we can't go into the pond. Local community needs are they want protocols compatible with traditional place-based values and practices, ability to conduct surveys with volunteers, flexibility due to evolving objectives, and fish left untouched and undisturbed. This means no tagging or catching of any sorts of fish. The reality is Koloko is a huge and turbid fish pond. Fish are highly mobile and easily missed, and there are substantial environmental gradients within the pond. Next slide, please. So we have Ricky here going, I wonder if a density-based population estimate using a mean density generated using N-mixture modeling of data collected in a robust design, unreconciled dual observer survey would work. So with the parameters that we're giving, not being able to catch any fish or survey or go within the pond, it makes it kind of hard to actually have an accurate survey method to count as many fish, to get an accurate reading of how many fish are in the pond. So we're thinking of this method. Next slide, please. So our methods were, since it's a dual observer, volunteers are paired into two, given training on survey methods. They receive polarized sunglasses, a GPS unit, and a data sheet. They walk the perimeter of the pond in teams of two and stop at fixed survey stations. There are 30 fixed survey stations around the perimeter of the pond. They're given a 5-minute count of targeted species. So they stand at the spot and count for 5 minutes and count as many species of targeted fish that they see. And then after that, there's a 3-minute biodiversity count per station. After that, they record temperature, salinity, and dissolved oxygen, and they record turbidity as well for the visibility of the bottom of the pond. Next slide. Next slide, please. Oh, there we go. So our assumptions for our model, when one of the volunteers are at a survey station, we have the assumption that fish are no longer leaving or coming in within the pond. And since we can't count for the center of the pond, we're assuming that fish are no longer entering the center of the pond or leaving the center of the pond when the surveys are being counted. Fish are assumed to have a constant probability of being detected if present to at each station. So we're assuming that the fish are kind of in a stable place once we're counting fish. Next slide, please. So when we are moving, just previous one back. Okay, so when we're walking between stations, the model predicts that the pond is open, and meaning that fish are assumed to be moving freely within and without of the pond, and within and without of the center of the pond as well. Next slide. So we're using an N-mixture model for this, and that means that there are two graphs, and one of them is computing the number of individuals at each site. So and then the other is detecting the probability of individuals in a population. And then they're running that with their covariates as well, which is salinity, turbidity, and distance from the Makaha. Next slide. So our preliminary results are pretty promising based off of 6 survey events. We found a mean population density of ama is estimated to be 0.17 fish meters squared. So that gives us a mean estimate of 7,600 fish, 7,650 ama and koloko, with a local abundance positively influenced by the distance from the makaha. Ama also have a high detection probability, negatively influenced by salinity and turbidity. So you're most likely to see the ama further back in the pond. Next slide. So qualified success. Estimates and uncertainty seem reasonable given their approach. Estimates suggest about 1,700 fish per acre in Koloko. With intensive agriculture, we can achieve 5,700 and between 5,700 and 7,500 fish per acre. The next steps are determining repeatability and validity of the model assumptions. Methods have been approved by the community, but COVID has slowed adoption rates, and models were coded are and provided to the community, and we're providing training sessions planned for them as well so they can keep the data. Next slide. With that, I would like to give a big mahalo to all these people, especially the park services, for the funding as well. Thank you, and I'll take any questions.
All right. Thank you so much, Daniel. So I'll go ahead and kick us off here with a question. So I'm actually curious about kind of the recruitment of ama. Do you know how frequently recruitment is occurring, and is it mostly occurring from reefs that are nearby into Koloko and other fish ponds? Is that kind of how that works?
So for our models, we haven't been assuming for recruitment quite yet. We're just counting, we're not counting that as well. Of course, there's always recruitment going on, but we're assuming every fish that we're counting is a different species. So, well, a different individual. So when they come, we're counting that as well. But yeah, I'm not quite sure about where they're coming from recruitment.
Cool. All right, good to know. Yeah, we got a question here from Noah. Says, are you concerned about edge effects when surveying for fish on the perimeter?
The edge effects, I'm not quite sure what that exactly is, but we haven't been assuming for that as well. I'm not quite sure. I don't think I'm not too concerned with that, as we're not too concerned. Yeah.
All right, and we got another one here from says, great work, Nate. What are the challenges in teaching volunteers to consistently apply this method?
So yeah, there's the community really wanted this to be a volunteer base. So there's a lot of restoration for this fish pond. So it can be kind of difficult kind of teaching people, but we're always open. The park services are always open to accepting volunteers walking around the perimeter and giving survey methods teaching. Yeah, there is a lot of variability with people who actually are counting fish and detecting fish. So that is also accounted for in our model though.
All right, awesome. Well, again, mahalo. That was a great presentation, and thank you all for the great questions here. So we're going to move on to our last presenter here, who is Reed Test. And Reed, if you want to go ahead and take it away for us.
Aloha, everybody. Thanks, John, for the introduction. I really appreciate that. This is my project on the growth rates of Polychaeta meandrina cauliflower coral fragments on different substrata. Next slide, please. Corals, in particular, Polychaeta meandrina, build complex structures that increase biodiversity among reef systems. These then form niches where organisms can live. Coral reefs play a beneficial role in coastal protection by diffusing wave energy. Polychaeta meandrina in the picture to the right is found in maximum wave height areas around Hawaii. Next slide, please. Coral health in Hawaii and around the world is seen decrease due to bleaching events. The following are some of the reasons for unhealthy reefs: coastal pollution, sea temperature increase, sea level rise, and ocean acidification. Due to all of these stressors, better methods of propagation are needed. Next slide, please. My project's main goal is to focus on the possibility of an advantageous growing medium. Once an optimal substrate is selected, fragmentation of corals can be isolated to use this substrate. The fragments of the corals that are resilient to heat waves and pH changes can also be placed on this substrate for successful future propagations. Next slide, please. These are some of my hypotheses. The first one is there a difference in growth rates among different substrata? The second is, do coral fragments use the same skeletal structure or grow over the substrata? Next slide, please. The first objective in starting my project was to maintain a coral colony to fragment. Thanks to the Mock Coral Nursery and Michelle down at PACRAC, I was able to complete that goal. The two colonies were then broken apart by hand, which increased the randomness. Each coral plug was then randomly placed in the rack using a number generator in Excel. The substrate that the coral fragments were attached to consisted of concrete, coral skeleton, rope, and basalt. Each substrate had 6 replicates. The tank that you could see off to the right, super clean at the beginning. It's not like that now. The tank conditions were monitored and placed at healthy levels. Next slide, please. Coral fragments were superglued to the substrates for the fragmentation process. And to analyze growth, 3 types of measurements were used: linear, horizontal, and volumetric. The water quality was ensured by using a YSI, API test kit, and a refractometer. At the beginning of the experiment, I was testing weekly, but then switched to monthly due to time changes and difficulties. Next slide, please. Linear growth was measured using a caliper, and every measurement was taken from the same side of the plug to keep consistency. And as you can see in the picture to the left, I had notches on the side of the plug that I could tell where my last measurement was from. And then I would go from the base to the top of the longest linear growth branching. Horizontal growth used underwater photography that contained a ruler for scale. The camera had a ruler attached to the side of it so that the picture was taken from the same distance. And as you could see in the middle picture, it is a aerial shot. And again, I used notches in the side of the plug and compass, taking the measurements from the same side to keep consistency. And the last picture shows the volumetric growth used Archimedes principle. This consisted of weighing each plug while suspended in the water. Each growth rate was then put into the specific growth rate equation that considered duration of time. Next slide, please. These are my results for the next few slides. And within all this, my results for all the growth, a ANOVA test was ran for each of them, and it showed no significant difference for all growth rates. But over the 4 substrates, rope showed the most linear growth, as you can see in the graph with the blue rope. And the concrete actually showed the least amount of SPC growth, which is pretty interesting, and basalt had the least amount of variation. Next slide, please. These are my horizontal growth results with horizontal SPC on the percentage on the x-axis, and horizontal SPC growth had the highest values in the basalt substrate. The concrete substrate showed the lowest horizontal growth among all substrates once again. Next slide, please. These are my volumetric results, which are some of my most interesting results that I think about because of my second hypothesis question. And for this growth rate, skeletal substrate had the highest values, but showed the most variation. So further testing is going to be needed. And then again, concrete with lower values of growth rate. Next slide, please. Only one fragment of the basalt substrate died during this experiment and revealed that all substrates are viable options for a successful propagation of Polychaeta meandrina. The coral that died was also one of the smallest fragments, and that could have led to the mortality. By having multiple methods of measurement, I could further investigate into how Polychaeta meandrina grew on different mediums. This species of coral shows different growth rates on all the substrates that I selected, which is really interesting that it changes. To reiterate, the coral fragments on rope substrate grew more linearly, on basalt more horizontally, and skeletal showed more volumetric growth. An interesting finding in my study was that the concrete substrate had some of the lowest values of growth, and yet is used as a growing medium around the world for coral propagation. This leads me to wonder if we aren't giving corals the most advantageous growing medium possible. The findings for skeletal substrate may shed light on the idea that corals are reestablishing the coral calyx, but more data is needed for that with pictures and microscopes as well. More time is needed for all these conclusions to be verified because Polychaeta meandrina can only grow about 14 millimeters per year, as said by Pritchett 2015. Next slide, please. I want to give a big mahalo to the following people: Matt for putting up with me the whole 2 semesters, and Dr. McDermott helping me out a lot. She was really a big help. And then the following people, I couldn't have done it without you guys. It's really awesome that you helped me. And then lastly, just a mahalo to the Mop Coral Nursery for donating corals. I know that was probably not the best they give me corals. They might not come back, but I really appreciated it. Next slide, please. Mahalo for listening, and I'll take any questions that you guys have.
Awesome. Thank you for that great presentation, Reed. Yeah, I've got several burning questions. I'm a coral guy. I'm into this. First off, I was curious, were there any problems with algae or with having to deal with aptasia or anything in the tanks where you were growing them? Was it hard to grow them, I guess?
Yeah, so at the beginning, we did have some aptasia because it was within the Mop Coral Nursery, and I actually scrubbed it off and got rid of it pretty quickly. I did a lot of water changes, and so that was really nice to get rid of. The algal growth was actually a battle the whole semester. I got tweezers and I hand tweezered around each coral substrate so the corals could grow a little bit better, because I didn't have any fish or snails in there to help combat algae, and I didn't want that to be a confounding variable. But yeah, it definitely got dirty. This picture is first week clean.
So let's say that you were going to do the study moving forward, and you were able to, you were like, you know what, we want to bring these fish to try and maybe get a microcosm going or something like that, or a mesocosm. What kind of fish would you use or introduce, or what kind of other methods might you use to I don't know, deal with or mitigate some of those issues we were just talking about?
I think the biggest thing, I don't know about fish. I want to get more into that and the certain fish. But the biggest thing is I found out where water changes, just to battle that. I mean, snails would be nice just because I probably wouldn't have to get certified to have those in the tank. But I think fish would be a really awesome contribution to the experiment, because they would actually make a ecosystem, that's out in the wild, whereas, mine is a controlled experiment. So the results might vary because of that. But yeah, definitely want to do that in the future if I can.
Awesome, yeah. And we still have time for a few more questions and answers. So go ahead and throw them in the chat here. And I was also wondering too, have you thought about maybe trying to do something in the growing corals in the ocean simultaneously with growing them in the lab too, and kind of seeing if there's a difference there? It's super interesting to think about the structures, whether it's rope or whether it's coral being in a natural habitat versus in a tank too.
Yeah, I would love to do it out in the ocean. There's certain areas out there are a little bit easier for experiments, not as much wave action, but I think my results would be a little bit more honed in on like true reality values. This was kind of like like I didn't want to make it too complicated, first thesis. So I was like, okay, just do substrates and then go from there. But that's definitely for future research, because this sparked my interest for a lot of material on rejuvenescence, for where coral polyps will die and then reestablish within that polyp. So I mean that, I don't know if that happens maybe out in the environment. I'd like to see that a little bit more on my skeletal substrates. So yeah, definitely want to do both.
Yeah, but I think you have the right of it. Simple is better, right? Simple, start simple, build from there. It's going to get more complicated than you want it to be anyway.
Yeah.
All right, so let's see if we get any more questions coming in the next 30 seconds or so. And if not, then we'll start to wind down this session and break for lunch here. All right, so we just got a question here from Noah. Do you expect similar results with other Polychaeta species? And what what is special about cauliflower coral?
So I was reading some papers on the Polychaeta, and I think it's just a great species to study because of the speed in which it grows. I mean, it's one of the higher growing species. 14 millimeters is still, within the medium range of corals. So, I mean, studying other species of encrusting, I think would be pretty interesting as well, just because, they take advantage of the substrate more often than branching, where, branching probably, expends a lot of its energy going up. But basically, to answer the last bit of your question, I only had like 2 options down at the mop nursery. Michelle has some special corals down there that were like fluorescing, and she didn't want me to take those. So I kind of had like certain options, but they're a really hardy species, and it's great to study them because it's super hard to kill them. I almost did. I had a pH change all the way up to 9 right at the end there, and I'm looking at them, this week, last week, and they're starting to come back. So with a pH change of almost, I want to say like 0.8, they're, still just so hardy. So that was really great for my first experiment, just because I've never grown corals before. So I needed like a species like a grass or so that or like a succulent. You didn't have to water very often.
Yeah, and that's such a great note to end on too, is the idea that these things are still so resilient too if they're given a chance to recover, right? Even brought to sort of these really high pH gradients or really low pH gradients. Okay, awesome. Well, I think we're going to wrap it up here. So thanks for all the questions. Presenters, thank you all so much. You did such an excellent job today. So this is going to wrap up our first presenter session. We're going to take an hour break and we'll come back at 1:30 p.m. today. And when we come back, we'll have a really awesome workshop with Pat and Tangarō, who are going to be telling us about the connections between Hawaiian oli and birdsong. So we hope to all see you there. And again, big mahalo to everybody who's been here and participated. Also, really quick, just wanted to highlight the silent auction that is ongoing. We talked about it a little bit earlier in the day. You can follow these QR codes. There are some really amazing things that are up there, and a big mahalo to Darrian for putting it all together for us. So all of the proceeds are going to go towards club activities. And yeah, so please take a look at that, and we will see you at 1:30. Thanks, everyone.
Ule ule une na mahanu, hei nu vele hua pana iwa, hei nu vele hua pana iwa, ule ule una mahanu, hei ala, hei. I, aloha kako, and I am Taupouri Tangarō, and it is an honor to share this space with you.
Mahalo, and I am Patrick Hart, and I am also very honored to be here as well. I'm still trying to figure out the controls here, but yeah, great to see all of you, if not remotely. So, you know, I have been studying Hawaiian birds for the past 30 years or so, and birdsong for about the last 15 years. And Tangarō is a well-known hula practitioner, and mahalo Stephanie for that great introduction. He's also a chanter, a composer, and a Hawaiian cultural scholar. And he's been doing that for longer than I've been working with birds here in Hawaii. But you know, until recently, our two worlds ran in parallel, and I don't think they really overlapped a lot. And we've known each other for quite a while, but I think it was his wife, Kekuhi, that who first encouraged us to explore together the connections between Kanimanu, or the world of birdsong, and Oli Kanaka, or the world of Hawaiian chanting. Yeah, and so today we'll present for you what we see as the initial results of what we hope will be a long collaboration. So here we go. Well over, you know, 30 million years ago, the Hawaiian Islands first emerged from the middle of the Pacific Ocean. And the first sound that would have occurred would have been probably the sound of the waves crashing on the rocks, or the wind blowing on the rocks as the newly emerging islands. Over time, of course, they started getting populated by new species, such as the ohia lehua, that you can see here. Lots of resources for future animals that might arrive. And then all of a sudden, from over 5 million years ago, a flock of birds, very similar to this one, which is the rose finch, got blown off course from their homeland in Asia and landed in what we now know as the island of Kauai. And so some of the first songs of these birds would have sounded like something like this. That's not what I meant to do. Bear with me, please. So over the next 5 million years, that flock of bird evolved through this process of adaptive radiation into, you know, a minimum of 56 different species and subspecies, all endemic to the Hawaiian Islands. And as they evolved and into new species into all these new niches, each of them developed their own characteristic and unique kani, or sound. Yeah. And so by the time just before humans arrived, and for a long time after that, the land was just filled with the sounds of birds, and very other animals were making sounds, because mostly what we had in Hawaii was birds and nothing else, right? And so it was just incredible, as Tangarō chanted, ule uleu, or just the chorus of birds in the forest. And it would have sounded something like. Soon after, the first Polynesians arrived, bringing with them the mele and the kani from their lands as they settled in Hawaii. And this is a painting of a, early of a Hawaiian village, and the mele, or the songs of these Hawaiians surely blossomed as they interacted with their new landscape. Yeah, that was basically, they're just surrounded by the sounds of all of these birds. And so birds and their song they were everywhere. And so Tangarō and I, really believe that the mele of the birds really informed the way the new human mele would grow and develop. Yeah, and so over time, the forest receded from the coastline as humans practiced agriculture and lived off of the land. And so the birds started to become separate a little bit more from where the humans lived. And so to a certain degree, this broke the connection between birds and people over time. And part of what we hope to do today, in some small sense, is to reestablish this connection between birds and people. But the early settlements would have had a lot of nesting seabirds that would have sounded something like this. So, many of our Hawaiian birds have been lost over the centuries, but we still have many left. And often heard is the uaukani. This is the kani, or the sound of the night. Yeah, many of these seabirds sing at night.
Patrick, may I jump in here?
Yeah.
May I intersect here? Aloha kako, and again, thank you for joining us. And I think it's so we're born into a world of hula, born into a world of chanting, and oftentimes we receive hula and we receive chanting through instruction, and great moments, we understand what it is that we're doing, but we understand the lyrics of the song, the history, the language, but what we didn't really pay too much attention to is where are the melodies coming from? What are the influences on how we present our chanting voice? And so I think when Pat and I got together, and it hit me when I realized that when I go to the mountains and what I hear there, on Saddle Road or in Volcano Village, that bird sound, that was once a very, very common sound down near the shoreline. And I'm thinking, wow, how would have that influenced our chanting if that was a prominent sound? Now, I do know that in at least the traditions of Halau Kekuhi, that we are trained to use our voices to imitate the lava. We call that aiha'a, the very bombastic moving of the earth. And so when we chant with all that vibrato. So we imitate that. We also know that sometimes we're taken to the ocean, listen to the ocean and begin to chant, within the currents with the breaking waves and imitate that. So it's endemic to our chant practice to mimic the world around us. Now, as it comes to animals, we have pig dances. We don't have too much diversity in animals in Hawaii. We have a pig and a dog and a whole lot of birds and a whole lot of, ocean people. But we have pigs and we have pig dances, and pig chants. And in those pig chances, we imitate the sound of the pig. And if I had, a full costume on, I could do the dance for you and I could do the chant for you. It has to do with fertility. Then we have the dog dances. And what do we do with the dog dances? We imitate the sound of the dog. So imitating our world around us is nothing new. But what was absent for a while, and I think this is the time when it's re-emerging, is reacquainting ourselves with the sounds of the birds. And firstly, seeing what are we doing today with our voices that we could align to what we think is similar to the birds around us. So I think as we begin to look at the different sounds of these selected birds, what we're going to do is we're going to show you how a chanter begins to make connections with these sounds. And again, it is nothing new for hula people to listen to the wind, and then we begin to move with the wind. So hula is really a dance of nature. How our body moves come from nature, and how our voices sound comes from nature around us. But what we're exploring is perhaps what do we have from the birds, and closing the gap between the birds and what it is that we're doing today. So you're part of an interesting exploration. And I think we have more questions than we have answers, but who knows, that's always a good way to start. But we're going to be going through the birds here. And Pat will be talking a little about the birds and giving you the sound of the birds. And then as a chanter, I'll begin to throw in some of my take on that. And hopefully we can get informed by this process. Okay.
Yeah, thank you. Yeah, so, one of the first birds we wanted to present for you was the uaukani, as I mentioned, and it would have been one of the ones that stayed around, and you can still hear them down in the lowlands. This bird is still living with us, nesting in some places, and I know Tangarō has heard this underneath his house on Oahu, yeah, at or where you were staying one time in Black Point. And they're very susceptible to getting eaten by cats and mongoose. They go into their nests. It's called uaukani because this is the sound of the night in the lowlands, right? And even today it is. And so here, we'll just play it. It's very haunting.
So when Kikuhi and I were a guest of John DeMello, and he lived right on Black Point. That's right, I guess, near Diamond Head. It's right on the point, right on the beach, and we're sleeping. And under our bedroom, we heard these noises. And I come from the mountains. When you hear noises like this, you respond. And we never thought they were birds because I've never experienced birds like that. And we thought they were perhaps some kind of ancestors talking to us in their coded language. And then we're told that there were the birds that were nesting. And immediately, what do Kikuyi and I do? We begin to imitate them. We begin to hum it and try to fix our voice to the pattern of it. And lo and behold, that was a couple of years ago. I didn't know we were going to be in this presentation, but now I know that that's a natural thing for chanters to do, is to imitate that sound. But now, it would be great if we do the dance of the uaukani, then we begin to deliberately use the voice of it in how we set our vocal patterns. So, yay.
Yeah, thank you. Many Hawaiian birds are named after what they sound like. And the uau is one of those birds. So see if you think its sound sounds like uau.
And as a chanter, I would hear Now that would be the foundation of what could be possibly a very provocative dance. Na 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 na. And especially if we begin in not just take the sound, but take the character of the bird. Because we never separate the sound from the character. We put it all. So there's all these layers, these rich layers in a chant. And so it's important that we don't just use the sound, but we bring the character of the bird into the mele. And so it just adds a whole lot more dimension.
Right. Next bird, one of our favorites. Another one that we think, one of the reasons it's named Iivi is because when it sings, it sounds a bit like Iivi, as we'll go over later on in the talk today. But these are one of the birds that are, becoming endangered. They can only live in the uplands because of the threat of mosquito-borne disease. So it's becoming harder and harder to hear the song of the EEV. But here it is. See if you think it sounds like EEV.
So we know EEV, you know, that's one of the prominent birds, and we actually go up. And many of our chants talk about the EEV and apapane and some of these more, you know, the birds that we see most often. But what we never paid attention to, at least myself, into this merger with Pat and I is to exactly, so not just talk about, oh, there's the EEV and ule making their sound, but you know, how it is that we could incorporate their actual sound, or at least mimicking their vocal patterns in the chant itself. So I'll give you an example. I'm going to use the word auhea oi. Yeah, it's a phrase. Where are you? Where are you? Where are you? Perhaps I'm going to say auhea oi EEV. Where are you, the EEV bird? And then I'm going to now use this the vocal patterns of the EEV here, and see if I could match it. Okay, so can I hear that one again, Pat?
Sure.
Oh, Hawaii, oh, Hawaii, oh, Hawaii. So I would, and of course, you know, my job is not to sound like a professional bird thingy, but just use what I hear and begin to translate it. So I'm saying, where are you, the EEV? But I'm actually mimicking the voice of the EEV in this conversation that I'm having. And so that would be the kind, this is, so I'm showing you, I guess, the process by which chanters listen to the ambient environment and then translate it into chants.
Yeah, great. So the Hawaiʻi kepa, you know, one of my favorite birds, one of the only bright orange, the only solid orange birds in the world, lives primarily up at Hakalau above us here on Hawaii Island, right above the university, actually, at higher elevations. It requires old growth forest to live in, and it's endangered like many of our birds. It has a really high-pitched and very fast song, at least to my ears, which Tangarō is going to talk about some of the connections, but let me play the song for you. Stop it.
All righty. So this kepa is really interesting because we have this chant style that is called the kepa kepa. So it's non-melodic. It's sort of rapid. It doesn't have to be rapid, but it has a definite pattern. Da 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 or da 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 da. So when we listen to the kepa, it's natural for me to hear this kind of chant style. And again, akepa and kepa kepa, they sound similar. And in our Hawaiian learning, we never separate the two. We know that there is some kind of relationship. Now, there's this really famous chant, and it's called Holewai Mea Ika Ihia Kamakani for Kamehameha I. And it is done in a kepa kepa style. And I'm hoping that you're able to hear how a human would begin again to translate the akepa sounds into the oli kepa kepa. Can we hear the akepa again, please, Pat?
Sure. Yeah.
Okay, so there's a definite pattern, yeah, and so we'll translate it in chanting something like this. Hello, okay, and so that is the kepa kepa, and I'm hoping you're thinking, oh yeah, That sounds, I can hear it. I can hear it because our job is to want us to hear it. We need to have more people hearing it because there's no way we can get away from chanting. It's the world we live in. So why not learn who are the contributors to this culture of chanting? And the Akepa is a very clear, it's one of those easy alignments to make. Next, Manu.
Next, Manu. Right. Alala. So, oh boy, I think we all know a bit about alala, right? But our native Hawaiian crow, used to be very abundant everywhere on the Big Island, and now is extinct in the wild, only in captivity. And I'm going to talk a bit about that later on in our presentation here. But so many different growls and caws that are part of their repertoire. I'll play. Where did that go? I have here. Play some of it right now. Beautiful sounds of the forest. Sorry. Tangarō.
Okay, that's a hard act to follow.
Yeah.
Okay, so we have two kinds of alala that I actually learned as a student in Halau Kikui. One was the mimicking of the bird itself, the alarm system that the bird has, alerting people in the forest that there's some danger that's coming in. And so I'll demonstrate that in a bit. And the other is interesting. It's called the alala because of the opening of the prolonging of the vowels, the opening of the mouth in the chanting. So it alters the sound, and it is actually used to promote emotion. So it's often used in a lot of love chants as a way of getting the listener's ear to or the ear and the skin to tingle, and the skin to have the hair raise up. So they use the alala not too often, but they use it. And I'll demonstrate both kinds of alala. Now the first alala, we never felt comfortable doing it, and I think the reason why was we never heard an alala. So I'm speaking of my own experience. I learned this chant at age 19, and I've never heard an alala prior to that. And so when we're told to do this, we were like, oh, this sounds so weird. And so we loved the dance, but there was a part of the chant that just made us, we felt like really bad imposters. And it goes like this. I na aloha na kia manu molokai. I still have a difficult time doing it. And so we had to do that. And it was the only chant, but the only dance that we did that had that dramatic beginning. And then now that I'm listening to the alala, I'm thinking, oh, okay, yeah, it's not, I can hear the alarm, the distress. So here it is again. We inherit this ancient chant, and it comes with this particular voices, but the storyline behind those voices are missing. The ambient world from where these dances originate from, they're totally different. So we're recipients of it from a different time. So it's hard to make the connection to an alala when we've never heard an alala. But now my granddaughter can do the alala really well. In fact, sometimes when she passes my house, she'll just do these alala alarm calls. It just, she thinks it's so funny. But, so that's something I'm very fortunate that someone kept it alive. Even though it was odd and awkward and felt a little stupid doing it, they kept it alive, and they insisted that we do it. Because now that the alala is making a comeback, or at least we're becoming more aware of alala, then we're lucky that we kept this song alive. And so I leave that with you. Now the other alala is unlike the screams, and in fact, again, I'm going to use the word, I'm going to use the phrase "au hea oi," and I'm going to say the alala. Auhia oi ka'alala. Where are you, ka'alala? But I'm going to use the alala chant technique. Okay, and you're going to hear it. So this is what it sounds like without the alala. Oh yeah, alala. Yeah, or if I am to use a different kind of like, oh yeah, alala. Oh yeah, alala. Okay, that's without the alala. Now I'm going to put in the alala decorations. Oh, and so when we open, we prolong the vowels. That in a person that's initiated into the culture of chanting, that prolonging of the vowels is a way of getting the emotion to connect between the chanter and the person that's receiving the chant. So again, without the alala sound, oh yeah, alala. With the alala, oh yeah. Okay, and so we even have a chant style that it's called the alala. Yeah, oli alala. We imitating the alala sounds. And so, and I think that has to do with the alala's opening its mouth really wide and making those sounds. So in chanting, we open our mouths and we close it, open in our mouths. And so we need to investigate that further. But those are very strong. Again, a hula with the sound of the alala, and then the vocal decorations, the alala vocal decorations. That is not just about halaoke kui. This is kind of common in the world of chanting.
Yeah. Play it one more time.
Yeah. And Pat, maybe I should include that.
Yeah.
In the first chant, the chant that we're doing when there's the alarm of the alala. Now that chant is called Nakanikau o Nakia Manu o Molokai, the dirge of the bird catchers of Molokai. And it had to do with the time when the guns were being introduced, the bird collectors during the Victorian era. They came into the forest and they would capture the birds for their European collections. And so the alala would alarm the forest, but also they would bring their guns into the forest. And the story, they talk about the kaniakapu, just the hearing of the loud snap of the gun, the birds will fall dead from the trees. And so that's why it's called a kanikau, it's a dirge. And so the alala is likened to, that kind of alarm system that has to do with life and death alarm systems. It's not a time to sound pretty. It's time to it's not the moment you want to feel sexy, you use your sexy voice. It's like set the alarm. There's something coming in. There's something that's so it's an interesting thing now to learn the culture of the alala. And now we're making these connections. And so we want to thank those people that kept that song alive, because now we have a bridge to make some kind of connection with the alala.
Thank you. Yeah, that's definitely a sound that will wake you up. Okay, we'll go to the next bird here. Let's see here. Akiapola'au. So this one, another rare bird that lives in the forest above us here at Hilo, only maybe 1,500 of them left. We know it as the woodpecker of the Hawaiian honey creepers. It uses its lower bill to excavate holes in the branches of the koa trees mostly and pull out big old grubs from there. And, birders like me will often, its name is Akiapola'au, but when you hear its song in the forest, when I hear it anyways, and other folks that I know, to us, it sounds like, I can see you, but you can't see me. I can see you, but you can't see me. And that's one way we remember the song of the bird. It's so here's the song. Maybe it's, kind of loosely sounds like I can see you, but you can't see me.
So na 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 na. Now, interestingly, I don't hear I can see you. What is that? What do you hear, Pat?
I can see you, but you can't see me.
I can see
you're
you, but you
hiding
can't
from us. Yes.
so I can't hear that. But when Pat said that, that was quite interesting because we have this line that says pe'eoi pe'eo pe'eoiala. You hide, I hide, he hides. And it's in our hula pahu, it's in one of our hula pahu drum dances. And other hula schools do this same chant with the same lines, pe'eoi, pe'eo, pe'eoiala. And the interesting thing is the pattern in which it is delivered in the chant is very similar to this pattern. So right now I'm you know, I just made this connection. And so when he said, oh, the hiding, said, oh, we have a chant like that. And when I listen to the Akia Pola'au, I'm like, oh, I can even say that it's following the same pattern. So can we hear that Akia Pola'au? And then I'll follow up with the actual chant. So, and it means you hide, I hide, he hides. And na 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 na. And that is a really beautiful melody to build the whole chant around. And so, which is quite delicious, makes us think that, you know, we should spend, chanters should sit a whole lot more longer these days in the forest to hear these birds because it's very distinct. And what's happening, because we're creating chants outside of the ambient sound systems, sometimes and currently it's a concern that many of our chants are sounding the same. It's not, it doesn't reflect the diversity of the nature we live in. So, so yeah, and this Akia Pola'au is just quite beautiful. And I actually want to hear how it sounds pecking, because maybe that pecking would speak to its name, because for us, the aki means to nibble at, to peck at, and pola'au means, you know, to nibble at the log, to nibble at the tree. So, you know, I need to experience what that is like so I can go, oh yeah. And the great thing about our process is our process does not have to dismantle anybody else's process. We all kind of bring all of our own truths in connection to the table and have a conversation. But I love this beautiful sound, and I want to build a song just on the sound of the Akia Pola'au. And maybe I could even write a song about the character of that, and who knows, and bring that to life. That's very inspiring.
Well, I'm looking forward to that, Tangarō.
You're gonna dance it, buddy.
Yeah. Oh. And we got to get up to Hakalau and see that bird for sure, which we have not done yet. Soon though. But all right. Next bird, Elepaio. So, you know, many of us know this bird is just a friendly, curious bird that follows you around the forest. They like to come down low and, you know, check you out. Insectivore, you can see their little whiskers on the front of their bill there, and they seem to use that to sense the bugs nearby and help them catch the flies from the air. You know, canoe makers felt that if the Elepaio was spending time near a koa log, that log wasn't good. It might have is indication of there being bugs. Maybe the log was bad if the Elepaio was associating with it. Another thing that's really interesting about the Elepaio is we think of it as the sentinel bird in the forest. When you're in the forest, places like Hakalau, and a hawk, an io flies over, you know, they're predators on birds. And the first bird to notice the io is oftentimes the Elepaio, and it lets off an alarm call. It's like, and all the other birds hear that alarm call and they dive for cover into the bushes. So we think of it as like the sentinel bird in the forest. And a lot of that's because they're looking up for bugs when they forage, and all the other birds are looking down in the liko, in the leaves. And so the Elepaio has always got its head up. And so it's able to sense the io before the other ones. So here's what the Elepaio sounds like. One of the sounds.
So how I would translate that in chanting would be, oh here, oh, elepayo, elepayo, oh here, oh. Could you play that again? There's something that they're doing that.
Yeah.
On okay, got it. Oh here, oh, elepayo, elepayo, oh here, oh, elepayo, elepayo, iuka Halymenia, elepayo. And so you see, we can easily adapt that sound, that pattern into our chants. But the character, how would we use the character of the elepayo in our chanting culture? When Pat was explaining this to me, and you know how the elepayo sort of gives the, you know, the indication that that tree, that beautiful koa tree that might look perfect and straight, there might be some flaws, you know. In this case would be termites or something, some kind of bugs. And I'm thinking, you know, I have, you know, children, you know, and my children were at the point in their life where, you know, sometimes they would, you know, bring home a special friend, and you know, the friend looks just so perfect, but you know, we got to send out, you know, the sentinel guards to just check out this person, you know, and just to see if, you know, if this person is as intact as they're appearing to be. And you know, this is not just a Hawaiian thing. All peoples do this, but it would be very typical of us to write a chant about the elepayo, you know. And so it's not just the beautiful sound of the elepayo that we'll be talking about, but perhaps the characteristics of the elepayo to check out, to sort of assess. And so we have all of these chants that talk, you know, that talk about the human nature. But as Pat was describing the elepayo, I was thinking, oh yeah, there are moments when I could have write a chant about or I was the elepayo. I was sent out to go and check out, you know, the character of this person and sort of kind of report back. And so it's not just the sound of the bird, but also the characteristics that we can use as we defined or articulate our own human experience. How often do we have to be the elepayo and we have to be the sentinel while everybody's busy, you know, foraging? We're looking up, we're foraging, but in a different direction, and we have to do the sounding calls. So this is a lot of fodder for thought in this world of chanting.
And you were telling me about the word ele. Remember?
Oh, the elele, yeah, the word elele. Yeah. And you know, again, the elepayo is very similar to the word elele. And the word for us, elele, is to be a messenger. You know, the person whose job it is to, you know, to get the message across and back. So the elepayo can also, you know, the elepayo could be the sentinel person, but it also could be, you know, the person whose job it is to get messages back and forth. And so in that case, it would be a great character to be likened to, if you know, in chanting. So yeah, I like that. I just learned that as we're practicing this time around for this presentation. Yeah, the elele, I'm making that connection to the elele.
Yeah, you'd never guess we practiced.
Ah.
Okay, next bird. Apapane. So, you know, these birds, they drink nectar all day long. They're the most abundant bird left in our Hawaiian forests. They've adapted to, you know, drink the nectar from the ohia lehua trees. High energy, high pitched song. They're flitting about the forest. They're never still, very restless, always moving from flower to flower. And I think a lot of that, you know, comes through in their song, which sometimes they have, you know, we did some work on the Apapane song, and they have hundreds and hundreds of different syllables and song types that they sing, and it varies so much. But here's one right here.
Okay. So, you know, we're talking about the sort of the high energy high octane characteristic of the apapane. But interestingly, and this is where perhaps we can you know, if you have any mana'o on this, you can just put it in the chat. The name apapane, the word pane means to answer, and it could have other meanings, but one of them is to epanemai, to answer, to present the answer. The word apa means to be slow, to be delayed, and apapane means to be is a delayed reaction, a delay in speech. To have, it gives the idea of some hesitance. But it appears that the characteristic of the bird has nothing to do with hesitance or delayed speech. So, you know, it's a mystery. And I think we have to sit, I myself have to understand, you know, and be in the forest a little longer to understand, why did our ancient people apply the name apapane to this particular bird? Did they hear apapane in the way it made its sounds? Or you know, or it was a tease. Sometimes we get the opposite names, you know, if we're slow, they give us quick names, or if we're very quick, they give us slow names, and it's a kind of indigenous humor. But, you know, it's a great, you know, if you guys have any mana'o on possibly how does the name attach to the characteristics of the bird, or to the sound, you know, throw us a bone here, you know, let's have a conversation.
Yeah, you know, one thing I was thinking, Tangarō, is they do have one common call. It's like apapane, apapane, apapane. Well, it doesn't sound just like that, but you can kind of make it out. So maybe it's another one of those onomatopoeia type things.
Yeah. Then that would make all the sense.
Right.
And you know, that would make all the sense. But in the chanting culture, if I needed to describe, you know, someone that was a little lethargic in answering my emails, you know, they're flighty and they're all over the place, but you know, they're just not getting to me. I would love to apply the character of apapane to that kind of character, to that kind of a person where, you know, they're busy and they're doing all these things, but I just need them to give me one answer so I can move on. And they're a little delayed in answering my email or my text. I would call them an apapane. You know, it's a gracious bird, but you know, there's a little tease there. Yeah. You know, over here says in the text, it says that perhaps the elele could also be thought of as the roles of the middle seats in an outrigger canoe. Moving the message from the steerman and the stroker, the rest of the canoe. Beautiful. And I think the next time we have a conversation, we should sit down together and talk about these birds, and instead of just Pat and I having a conversation, we should widen the circle and bring all of these knowledge systems into that, because everybody holds a little piece of a truth, and we should amass that together. Thank you, SL.
Scott Larson.
Okay.
Okay, right.
Such a pleasure. Mahalo nui, guys.
Thanks, Scott. So hey, we're gonna shift gears just a little bit here. No, we're not going to do focus on the bird songs themselves, but another thing that we talked about in the abstract for this talk was just the way that some of the parallels in the way that the bird song has changed through time, and perhaps the way you know, human song has changed through human voice, human oli has changed through time in Hawaii. So sorry, bear with me. It looks like the next few slides are mostly me talking. So sorry about that. You know, I'll go through this quickly. So first, you know, when both humans and birds are making sounds, typically the sounds can be from, you know, gene derived, it's like nature, or memes, nurture. Yeah, so from learning. And so both birds and humans share that. And so birds, like humans, need to sing or create sounds to communicate a lot of things: territoriality, you know, attracting mates, talk about avoiding bad things or predators, you know, keep the group together, sharing knowledge. So a lot of parallels in the reasons why birds and humans would communicate acoustically, right? So I just wanted to, you know, talk just a little bit of work that some of our students have done, just looking at at some of the way song has changed in Hawaii across the landscape. So some of you may know Joshua Peng Ching, who's a grad student a long time ago, 10 years ago now. And he found that, there's these cultural traits in Amakihi that have become established across the landscape where they sing a different song just over really short geographical areas, like in Lower Puna. You can see right here, all of these spectrograms are typical of Amakihi from that region, and they don't overlap. So they've almost learned dialects over just really short spatial scales, yeah? And also, we talked about the alala, Tangarō, and I were talking about that earlier, and they're one of the most intelligent birds in the world. They came here more than 1 million years ago. Their ancestors, they have this really rich, culturally transmitted language. And when they went extinct in the wild, they shared this language among all of their, their whole population. They shared this rich language, and they went extinct in the wild and were brought into captivity. And so, one thing that we looked at in our lab, Ann Tanimoto Johnson, a graduate student in our lab, looked at how changing from a wild population to being captive, how did that affect their vocal repertoire and their vocal behavior? How did their language culture change from this? And so, she found that they have all these different behavioral categories of the things that they say to each other: alarm calls, broadcast calls, aggressive scolding calls. And she compared some remaining tapes that were given to us from Paul Banco and others of the last wild alala, what they were saying to each other, to what the remaining captive alala were saying to each other. I wanted to see how their vocal culture had changed. Yeah, what have they lost or gained or whatever? And so you can see from this slide here that, for example, if you just look at alarm calls, the wild birds had all these different types of alarm calls, and the aviary birds had different ones, but there wasn't a lot of overlap in them. So their vocal culture had changed over time from the wild to being in captive, right? And Anne found that there was even these calls that had been completely lost, like we had from these old tapes. And Tangarō is going to speak to this a little bit, the parallels in chanting in a second. But the wild birds had all these different broadcast calls that they regularly gave that were no longer ever heard in the birds in captivity. And so and because they're culturally transmitted, they learn from each other. We think that they were lost completely from their populations. And so I'll just play a couple of these. No, those aren't the ones. I'll play a couple of these lost broadcast calls from the wild birds to the captive birds. So here's one of them right here. So that's something that you just don't hear the alala say anymore. And here's another one. Oh, didn't mean to do that. Having some trouble with my controls here. So, Tangarō, there's you're up.
Yeah. All righty. So okay, so we have 3 chants, and we're going to play, but because of time, and we want to actually teach you a chant, so we want to reserve enough time. We're going to just focus on this middle one to provide us some kind of audible example of the evolution of voice in the human realm. And so the first chant you're going to be listening to was, or the chant you're going to be listening to was chanted by someone that was born in the 1800s, and it was recorded late 1800s, early 1900s. And so this is it. His first language is Hawaiian. Yeah, that was his primary language. He was reared in a culture of chanting, of his whole world was very, very Hawaiian. Okay. And now these are variables. So he in his waking hours it was Hawaiian, in his sleeping hours it was Hawaiian, and everything was Hawaiian about him. Hawaii at that time was opening up to the world, so there were these foreign things that came into it. But this is what a person of that generation sounded like chanting. And can we have that, the middle one? Yes. Okay, so this is, and again, at the time this person was born, we can assume that the forest was a whole lot more closer to the shoreline. Yeah, there are larger tracts of forests that served as bridges from the mountains to the shoreline. So this would have been, you know, we could assume that this chant was, he assumed this chant during a time when the bird, you know, the ambient sounds are very, very healthy. Now, this is the same chant as I've learned it in 1980. So some of you might be thinking, oh, 1980, that's like 100 years ago. But that's when I was at college in the university here at Hilo. But this is the same chant. Now, what I want you to do in the chat is speak to what's the similarities, what's the differences, and what are some of those possible conditions or variables that make it sound the way it does. Okay, so just put it in your chat. And here it goes. Okay. O pele o ke kumu ka hiki na na I hola le te po aku kanike ku ike ka hiti te mo tu neva ula. Ila e tōpili atu e tōpili alo e tōpili noho ali I ka uere I ka lani nehe I ka honua kohia e ka ne. Rohi e tani te motu wahi o wai ka I ka lani na pele I ahi ke ane ki lau e. O waka ka niho te atu ai o hiko o hau mea te aku. Hele ai ako me ka hale o pele wai noho eli te una. Okay, I hope you heard differences. And what were some of the differences you think you may have heard? And what would be some of the, again, the variables that made for those differences? Okay. I'm waiting for you to add a chat something.
Yours was more melodious.
Okay, more melodious. Okay. And what would be possibly the reason for that, more melodious versus you know, monotonal?
Good question.
So I want you to think about that. Of course, we're trained to, you know, if we have an old recording, we train ourselves to sound like that old recording. So we can imitate this old man that has died 100+ years ago. But, you know, how we actually learned it. And if you go to Metabolomics just done, and you listen to what the chanters used to sound like 120 years ago to how chanters sound today, you're going to hear that there is a difference. And we want to begin questioning what are the variables that causes those differences? And then how do we grow back? What do we need to create to return to, so if we want to imitate the bird sounds, you know, that's one thing to imitate them, but to understand what they're actually saying requires that we become quite intimate again with those spaces. So I want you to think about it, and you know, and understand that the bird, you know, the evolution of the bird sounds are not very different from the evolution of the human sounds. And because the human being, and interesting, the more urbanized we get, how does that affect our chanting styles? The more removed from nature we get, and we get centered in the urbanization, how does that affect how we sound? Okay, we have Scott here. We imitate human vocalizations, not the origins of more than human vocalizations linguistics, okay? Okay.
Yeah. Very true.
Thank you. Tangarō, have you experienced a change in your own practice of hula or the way you sound after spending time in nature? Yeah. Oh, yeah, it affects how I dance. And that's interesting because we all do know that hula is a nature dance. It originates, we have many origins of hula. So there's not just one school of hula, many origins, but they all come from nature. And interestingly, when we learn hula, we go into a hula studio. And then later on, there was called the hula halau, which is really a room with, space. And we begin to learn hula from a human being and a human being. So that's a way of learning hula. And that's a good way. But when you actually go out into the forest and learn from nature directly, there's a different sensation. And so, yeah, I think that's part of what we do nowadays. We don't just teach our hula people hula in the room. We got to take them into nature and teach them hula, so that we tell them to imitate the alala. They don't feel awkward doing it because they've never heard it, they've never seen it. Okay, we imitate kids. So yes, it does it has a huge impact when I'm in the real world. Pat Perez says the older chant sounded like it was more head voice and yours was more chest voice. Perfect. Now, what are the variables? Why did 100 years ago, and interestingly, we have like 20 recordings of people in that same time period, and they all use this we call it nonolo, a very light head voice. But then 120 years removed, the voice gets a little bit more deeper. And so we want to be able to understand why that is. 100 years from now, our voices coming from our toes is going to get deeper as we get removed from our nature, or Obvious is the older chanter head voice. The older chanter was slower with more pauses, while yours was faster and more hurried. Yeah. That has nothing to do with the chat style. That has to do with trying to save time at the end of this the end of this presentation, so we can teach you a chant. So you got to forgive me on that one. Okay, let's move on.
Good segue. That's a good segue.
Yeah.
There's one from Drew, real fast.
Okay. Drew, the older chant had a distinctly fluid sound, and the one learned in the 1980s had more clear enunciation. Beautiful, beautiful. In the 1980s, what was happening? We were not native speakers. Well, I wasn't a native speaker, and most of us in school at that time were coming to college to reclaim our native language, to revitalize the native language. So we were learning Hawaiian language from a linguistics, an academic linguistics platform versus the domestic. And so we had to enunciate, our okinas had to be in place. When we become a second language speaker, we approach the language a whole lot more fanatical. So enunciation and make sure that, that all of it is there. But you can tell a native speaker, because a native speaker, we just kind of bust up the language. When we talk about English, when we look at English, the people that learn English, they speak it so well. But the people that are born into English families, at least in Hawaii,
True.
The British people are trying to still figure out what it is that we're saying. A good point, Drew.
Good point. Hey, so I am going to, in the interest of time, it looks like it's 2:33 right now. I'm going to skip the next couple slides, which is, I think, okay. And we can get into, well, if that works for you, Tangarō, is mostly just talking about some other studies that we've done in our lab. We want to talk about the ho'okupu for the birds, things that we can do for the birds. I think we all in the TCBES program know, ways to help our birds out these days. One thing that we're talking a bit about today is learn the bird language. a little bit as we've been talking. But we wanted to get to this last slide. We wanted a little bit of time for this. And so this is a good chance for, I did not mean to do that. Did not mean to do
It's
that.
alarming. It's not you're scaring our attendees away with that one. They're saying, jump ship, just end this call.
I know. Oh. Tangarō, would you like to?
Yes. So
yeah.
Part of one of the very, very personal reasons why Pat and I agreed to work together, and one, it's an easy working together, but you know, we have our personal and professional missions, and most of you know Pat, and the introduction to spoke of how he may have influenced a number of you through the academic corridors, the academia, and then here I come in with the cultural piece. And you know, one of my concerns is now, how do we become fluent in bird language again? How you know, and for many of us, the birds are ancestral. Many of our families, their ancestors will, upon death, will assume, or they'll transfigure themselves into some of the birds. So it's deep, and it's very, very personal. It's family. And so how do we get our people, our community, to become acquainted again, so that we're not just doing a poor job of kind of glossing over the bird things in our chant. Everything that is about bird, we're glossing over it because we don't know nothing about it, when the birds are right up the road here, and we should do something about it. So I'm coming to it with this idea to revitalize our bird language, at least for humans, you know, our human ability to recognize the birds and recognize what it is that they're communicating. We had to revitalize our Hawaiian language. We're down to 2,000 native speakers in the 1980s. And now we're 20,000 plus speakers of Hawaiian language. So it's something that we can do. And so I created this little song here, and you know, it's a way of just getting our children, either our children or the child in us, to begin to investigate and mimicking bird sounds, at least the ones in Hakalau. You know, so here's this little ditty. It's a little ditty, and it says, "Ahakalau ahaka lehua o manu ule ule punana," and then we say he iivi, you know, the name of the bird, and we plant that in and say pene. This is what the iivi bird sounds like. And then we do the chant again, and then somebody will call out the elepaio, and then somebody would jump in and say, this is what so it's a game. And then we can even switch it around. After a while, we can do the bird sound and then ask the people to guess what bird it is. But you know, it's a way of getting us closer to our bird people, and learning again from them. And then also becoming aware of their habitat and the concerns. Because once we begin to value something, then we're going to protect it. And right now, it's hard to protect something we don't have any connection to. So part of it is building a connection, you know, beginning for, you know, and I'm sure all of us on this call have some kind of connection and are doing things already, but the majority of our Hawaiʻi is not. And so things like this, working through the charter schools and getting these kinds of curriculums out there and getting these kinds of concerns out there. I mean, we do it for our whale people, our ocean people. We can do it for our bird people. And so, so this is how it sounds. And so I'm going to chant it, and I'm going to call the bird He'i'iwi, and then Pat is going to mimic the I'iwi sounds, and you can have a lot of fun with that. Okay, and be prepared. Some of you have a need to jump in there. You know, just unmute yourselves and jump in.
Feel free, everyone. And I am not going to mimic it, but I'll do a very poor job of mimicking the I'iwi. Let's just say that.
Okay, so he's gonna just plug in. I guess you're gonna just put the sound in there. But what we do want to do is eventually get the human sound. We just read through this yesterday, so he's a little nervous.
I can try to mimic it, and then I'll play what each one sounds like. How's that?
That would be perfect. Okay, ready?
Sorry, everybody. Here's what
Okay,
they
can we
really
hear
sound
the
like.
can we hear the first language?
Oh,
That's the indigenous speaker, and then here we are. We're part of the revitalization team. Okay, and so we're going to be doing the Elepaio next. Okay.
okay.
Ahakalau, ahakalau, ahakalehua manu, ule ule upuhuna na he Elepaio pene.
Lepaio, 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 lepaio.
Okay.
Here it is. I know. Okay, so they do it better than I do. Let's just
That's their native. That's their native language. We're just, you know. And so, do we have any brave souls in this call that would like to do the ua'u bird? Or you can choose any one of these birds. I'll just chant it for you. What is your favorite bird? Anyone? Anyone? Just raise your hand. Don't be shy. You can just put it all on Pat, and you can.
Ua'u, ua'u.
Drew, do you want to do one of those birds? No? Okay. Well, Pat, this is your ua'u, Pat. This is like your signature bird. This is your signature bird. This is your signature call. Make it work now. Ahakala ua'u, ahaklehua umanu, ule ule upuhuna nahe ua'u pene.
Ua'u, 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 ua'u. It's ua'u. Here's the real version.
The native speaker. No. You didn't do the Pat.
I didn't. Yeah.
Oh, you got to go back to 101. What 101? Okay. And then
could be worse.
We're gonna do our alala. Ahakalau, ahakalehua umanu, ule ule upuhuna nahe alala pene. Wow, 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 wow. Pat, you know, that one sounded like our cats eating in our next door neighbor's yard, but you can work on the alala. I like your ua'u, Pat. I can work. I like your ua'u, but maybe remedial class for alala.
We've got to get Kekuhi to do the alala. She's perfect.
Oh my Lord, no,
Kekuhi
she acted out. She takes it too seriously. She's intimidating. It's scary. But anyway, guys, why don't we do some Hawaiian language things here? We have 2 minutes. Can you just repeat after me? Because I imagine that you're going to use this chant. Yeah, whether for yourself or, and then if you want to investigate more, you know, you can always email me and we can set up some time to just get this going. But it's a fun way. Wouldn't it be fun also to just do the calls and let somebody else guess what it is? The alala. Oh, that's like a cat. I'm only teasing Pat. Okay, so repeat after me. Ahakalauau. Ahakalauau. Ahakalehua o manu. Ahakalehua o manu. Ahakalauau. Ahakalehua o manu. Ule uleu punana. Ule uleu punana. And then it's he and pene. Pene is like this. This is what they sound like. Okay, so let's chant this together and we're going to do the ua'u. Pat, we're going to end on the ua'u. Okay?
Okay.
So we'll all chant together and I will do it really slow. Okay? Ahakalau wau. Ahakalehua o manu. Ule ule upu nana he. Uwa upene.
Uwa, 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 u.
Yay! Okay,
Yay.
I think we've arrived. So guys, this is for you to use.
There's still people online. They haven't all left. Great.
Yeah. Well, you know, after you did your alala thing, they want to know what else is there that we can, you know, just lighten the load and have a nice humorous laugh to it. But, you know, guys, this is part of it. And learning any language is risky, you know, and it does take, you know, it does put us in vulnerable spaces. I remember at one time, we didn't want to do Hawaiian things because we didn't want to sound odd. We didn't want to sound like an outsider. But that's the initial steps. So how do we encourage people to learn the bird calls? And for what purpose? And so we leave those questions and answers to all of us. We need to figure that all out. But in closing, I want to just say this conversation has added a lot of fuel to my creativity, my inquiry, my being curious. And no longer am I satisfied just glossing over, oh, they're talking about Iivi or up upon it. Now it's every time I do a chant, all those chants that I do that touch on birds and the environments of birds, it's making me want to go and figure this out. So I want to say thank you to all of you, and especially to Pat for adding to my hula. And you know, and through hula I touch thousands of people. So your knowledge is not just your experience. This experience is not just for me here. It will radiate out and maybe get other people to look at their birds in their indigenous spaces, and let's grow a concern. And that's my closing to you, Pat.
And likewise, Tangarō, I mean, working with you, it's expanding what I do in my science. It's expanding that. So I just think I'm just really looking forward to just continued working with you and collaborating in this area. It's been fun.
Yay.
So mahalo for that. And thank you everybody for coming.
Again, if you have any questions, do email Pat and I. And let's grow this together. Let's jump in this together. And let's fill up the seats of the canoe, to use that metaphor. Let's fill up the seats of the canoe and let's inform each other on this journey toward creating healthy bird systems and environments, and making room for chant and hula and the arts to participate in this movement that is utterly mostly science. The cultural piece should come in there also because it comes with its own community, and we all need to do it together. Thank you very much.
Thank you so much. What a fun way to come back and awake after lunch. And I think we all need to practice and gain some courage on our bird songs, but we got some good practice in there. So thanks for showing us how to do it. And yeah, what a beautiful project and collaboration. And yeah, I hope that we can find the space to continue to practice and learn the songs together. So thanks everyone for participating. And yeah, if anyone has anything to say too, feel free to unmute and contribute. I think we'll be switching.
Yeah, we got to move over soon, right?
Yeah. We'll do the tech on this end, but if anybody wants to unmute and say anything, feel free. Thank you so much.
Thanks. It was fun.